Ito yung dito natin pag-uusapan kung ano-ano ba ang mga kailangan ng mga mikrobyo. Okay? Alright? Dito natin pag-uusapan kung ano-ano ang mga kailangan ng mga mikrobyo para tumubo sila. Specifically, mga bacteria. Okay? Somebody is coming into my shot. Inaagaw ang limelight ko, guys. Sorry. Alright? Okay, so let's start first. Let me ask the question first. Why do medical technologists need to know the nutritional requirements of microorganisms, to be specific, bacteria? Who can answer? You can guess, don't worry. I won't bite. Somebody raised his hand. Okay, Nigel. Why is it important for a medical
Dah,
technologist?
dahil nagko-culture po tayo ng bacteria.
Okay. All right. That's a very vague answer. All right. Thank you so much. I love it. It's vague, but it's straight to the point because we're culturing microorganisms. That's good. What about another person? What about McKay? Where's McKay?
Hello, sir. Kasi, sir, meron po mga bacteria na need ng additional requirements para mag-grow sila.
Very good. That is actually one of the things that I'm asking for. They need additional requirements. They are what we call fastidious. Some organisms are fastidious. So yun yung mga maarte. All right, so we're going to talk about them in general here. I prepared a lecture. I prepared this lecture in such a way that I'm asking questions. Okay, so if you're not familiar with my teaching methodologies, do we have new people here in the classroom? If you're not familiar with my teaching methodologies, I usually ask questions before we actually go on to the deeper discussion of the specific topics. Okay, so that's my teaching methodology. So feel free to answer whilst you are listening in on me. Bayaan nyo lang ako magdaldal-daldal dito. O whilst also participating, okay? And remember, Deandra won 100 Gcash points from me yesterday. I think I sent her money. Deandra, did you get the money? Because alam niyo naman ako, ang inyong butihing lecturer, hindi lang nagtuturo, nagbibigay din ng pera. Di ba? Para iahon ng mga medtech sa Pilipinas. All right. So, let's start that. Thank you so much, Nigel, and McKay for that wonderful introduction to the topic that we're going to discuss in today's lecture. So, let's start first with differentiate between a lithotroph and a heterotroph. This is basic biology biology 101 back actually. So, a lithotroph is an organism that relies on inorganic substances, whilst a heterotroph relies on organic substances. Usually, these include elements. The most important thing that you need to remember is that two terms refer to carbon as the nutrient, as the main nutrient for requirement. So organisms that thrive on inorganic substances are lithotrophs, and a heterotroph is an organism that relies on organic substances. The two terms refer to carbon as a nutritional requirement for the growth of microorganisms, specifically bacteria. Okay? Now, what element is required for bacterial protein, RNA, and DNA metabolism? This is very important, by the way. Even humans or eukaryotic cells of eukaryotic organisms must have this. Otherwise, there is no life. Who can answer this one? What element is required? So we talked about carbon when it comes to lithotripsy and heterotrypsy. What about element for protein, RNA, and DNA metabolism? Somebody raised his or her hand. Let's start with Alexis. Alexis, why are you not showing your face? I need people to show their faces.
Sorry, sir. Medyo poor ako ngayon kasi naka-hotspot lang
Oh
ako.
my gosh! Girl! Parang kailangan ata nating mag-send ng 1K Gcash para lang kay Alexis next week. Okay.
True, sir.
Alright, so what's your answer?
Nitrogen po.
Nitrogen, very good. Nitrogen. That's why in clinical chemistry you have non-protein nitrogenous substances, right? These are metabolites basically. Okay? Can you guys see the lecture sorry, can you guys see the lecture in the shared screen? Please let me know, Mark.
Yes,
Okay?
sir.
All right. Thank you. What about in our YouTube in our YouTube video?
Yes po.
It's okay. It's okay. It's functioning. Okay, very good. Thank you so much. All right, let's move on to the next question. So, examples of minerals required by bacteria. So, what minerals do we need? Again, these are examples of compounds. Sorry, not compounds. Basically, what do you call this? They are, let's say, metals. I referred to it as minerals, so it's easy for you guys to remember because when you're drinking your vitamins, you see minerals such as magnesium and sulfur in one of those. Sorry, you see magnesium is as one of the ingredients of your multivitamins, like zinc, right? So in this case, microorganisms require bacteria in general require magnesium and sulfur. Magnesium is important, remember, because last time we talked about it, right? It goes in and out of the cell, right, of the bacterial cell. Okay, while sulfur is actually something that we will talk about when we discuss specific topics on Enterobacteriaceae. Okay, so you guys remember your Enterobacteriaceae organisms? Enterobacteriaceae. Okay. Alam ko sa Pilipinas, ang base sa kanya, Enterobacteriaceae. Alright? But the correct pronunciation is Enterobacteriaceae. Okay, so who here remembers their organism or their Enterobacteriaceae organisms? Let's see. I'll go one by one. Let's start with Arnie Salan because you're in the middle of my chat room. Arnie Salan, give me an example of an organism belonging to the family Enterobacteriaceae.
Salmonella po.
Okay, very good. All right. What about Clarice Solabo? Because she looks like she's dozing off. Clarice.
Yes po, sorry.
Ano?
Ano po, Shigella.
Alright, Salmonella, Shigella ba? Parang lahat ng students ngayon gusto talaga ng mayroong mga highly pathogenic organisms. Pwede ba tayo pwedeng maghanap ng ano? Okay, what about Crisia cinereus? Because you're also in the middle of my chat. What about Crisia? Dai, nakamute ka, Dai. Charing. What's that? What's your answer? Give me an answer. There's so many, but I'm looking.
Ano po, Klebsiella po.
Okay. Students really love pathogenic organisms. They really love pathogenic organisms. Okay. What about our dear moderator, Mr. Mark Segi? Can you give me an example of an organism belonging to the family Enterobacteriaceae? Last na to. We're going to move on na after ni Mark. Syempre baka mamaya sabihin hindi ko pinapansin si Mark.
Nakalimutan ko, sir. Pass muna po.
Pass? Hoy, Enterobacteriaceae. Magagalit talaga sa inyo si Sir Marco, ha? Kayo talaga, guys, ha? Alright. So, thank you so much for participating. Pass daw muna si Mark Segi. So, tulungan ko siya. I have a feeling that his answer is actually Escherichia coli. Okay, E. coli lang. Hoy! Mark, ha? Sabalik kita, Mark, ha? Nakakainis ka. Hindi porket moderator kita, Mark, ha? Hindi ka na mag-aaral. Hoy, Mark, ha? Alright, so thank you so much, people, for participating in our mini recitation. Okay? Now, what is the term for salt-requiring organism? Oh, I love this because you're going to hear this when we talk about a specific group of microorganisms. The term for salt-requiring organisms. Okay, Valiant. Ayan na! Minty! Ayan na! I love it! Ano la? Itaas ang bandera natin.
Halophilic, sir.
Mali ang pronunciation, pero sige lang, go. Alright, I'll accept it. It's halophilic. Alright, wonderful. Thank you so much. Halophilic or halophilic, it doesn't matter how you pronounce it as long as you select the correct the correct answer. All right, thank you so much. So give me examples of organisms that are halophilic. Anybody in the chat? Alexis again.
Vibrio species
Okay,
po.
what else? Oh, thank you so much. Wonderful, very good. What about Maria, Christina, McKay?
Staphylococcus po.
Staphylococcus. Very good. You guys are reviewing. I love it. These are general questions that you should know. All right, Vibrio species and Staphylococcus. All right. Actually, we have what you call MSA, mannitol salt agar, that allows the growth of Staphylococcus only. Selectively grows Staphylococcus or cultures Staphylococcus and not other microorganisms. I love the at home atmosphere in Valiant's room. I love it. It's so cozy and everything. I wish I had that kind of setup when I was a kid. Okay? Not saying I'm still not a, I'm still young at heart. Okay. Let's move on. Okay, I'm 30-ish. By the way, guys, I'm 30-ish. All right. So, which species of Vibrio is not considered a halophile? So, we're going to go a little bit of a discussion on Vibrio so that you guys remember. All species of Vibrio or Vibrio, according to some of my professors before, Vibrio, okay, that are not considered as halophiles is Vibrio cholerae and Vibrio mimicus. So these are the only organisms that are not the only Vibrio species that are not halophilic in nature. Okay, so all species of Vibrio are halophilic except for these two. Okay, so you'll have a chance. Well, you'll have a chance to revisit this when we talk about Vibrio in our future discussions. All right, now Mycobacterium tuberculosis is an Mycobacterium is an organism. Okay, let's put a mark on this one. All right. Is an organism that requires an increased concentration of what? Okay, so another growth requirement. Mycobacterium is an organism that requires an increased concentration of what? Organism siya, okay? So remember guys, I typed this when I was still a student, so I didn't have that much. I was so worried about medical technology knowledge that I forego the grammar. Okay, so apologies for that one. Now, who can tell me the answer to the first bullet? Ooh, they forgot. Ooh. All right, Eliza, give it a try.
Increase of protein.
Very good. Wonderful. That's good. That's good. That's the reason why it's hard to cultivate MTB in the clinical laboratory. It usually requires like 30 days to read the LJ or the Lowenstein-Jensen agar. All right? It's a violet agar. Sorry, it's a green agar. It's a greenish agar that allows the growth of Mycobacterium species. Actually, not just Mycobacterium tuberculosis, but Mycobacterium species in general. But you'll remember the reason why I focused on tuberculosis is we have a short discussion on Mycobacterium species after our discussion of the general topics in microbiology. Okay, so the growth requirement is the reason why technologists use this as a component of culture media for this organism. What is the component? What is this particular component? Whole egg. All right. Traditionally, technologists found out that you need to add whole egg to cultivate Mycobacterium tuberculosis. Okay. So in today's world, we rarely use eggs because it's basically a cause of contamination. All right. But in the wee old years of microbiology, when they still didn't have the technology to sterilize and pasteurize certain components of agar, they relied on whole egg. Okay, so you might be asked the question in the board exam, because I remember one of my students before, he mentioned to me that, sir, what is the answer to this one? Select the following food products that is a requirement for MTB cultivation. So there's questions there that included chicken livers, All right. They said chicken livers. Anything related to food. Chicken livers, egg. All right. What else is there? Vegetable broth or vegetable mixes. I think it has something to do with vegetables. And I think the last one was the one that he answered, and it was the confusion. It was a confusing one. It's an extracts of yeast extracts. So it was a little bit confusing for him because what he would hear mostly in discussions in microbiology is about yeast extracts, blah blah blah. You would hear it when you talk about Legionella pneumophilia, the Legionella pneumophilia agar. Okay, okay. So charcoal Leyden usually has yeast extracts. So in that case, he was a little bit hesitant in answering whole egg, but he remembered. So that's the reason why I added this one here, this little one here, because sometimes you might be asked common sense or nonsensical questions in the exams. Sometimes it doesn't require you to think too much. All right, all you need to do is remember. Oh, okay, increased protein. Which among the following has increased protein? Vegetables, chicken livers, or mixed or yeast extracts. Of course, eggs usually have the highest protein content. So you might be asked that kind of question in our local board. So just remember this one, okay? So it's an uncommonly asked question. So I wouldn't say you should add a star to it, okay? What I would suggest you guys remember is the first bullet. This one. Number 73. If you're going to remember something, it's number 73. Okay? All right. Now let's move on to the next slide. In order to cultivate the growth of Haemophilus species, what is required? It requires what? There's two things that you need for Haemophilus species. Who can give me the answer for this one? Ooh, people are looking. People are looking. I think I should put a timer so it's easy for us. So, okay, Valiant. Okay. Minty Fresh.
Try lang, sir. X and V factor.
Wonderful. That's good. That's good. What are the other names for X and V factor? Who can answer that one? Thank you.
Okay,
Oh,
pala.
okay, you can answer again. Okay, sure. Sure. I'll
Okay.
give you the floor.
X factor, it's called hemin or hematine.
Very
And
good.
for V factor is nicotinamide.
NADPH lang, te. Huwag mo pahirapan na sarili mo. Okay? So sometimes you will be asked a set of questions. So, which of the following is specifically needed for the growth of Haemophilus species? Nicotinamide or what's the other one? Nicotinamide and X factor or hemin. They would give the choices, the other names in the choices. So please be mindful of these things. But in the laboratory, obviously we don't need in the clinical laboratory, it comes in packages of X and V factor. So you don't need to worry about it. Okay? So what it looks like, it's actually if you have you guys seen an antimicrobial disc? Have you guys seen it? Like a disc for Kirby-Bauer disc diffusion? No? Oh gosh, you guys really have no experience in the laboratory whatsoever? Oh my gosh. Really? Nobody knows what it looks like? Okay, so since we don't have any idea what it looks like, I'll Google search it for you guys and I'll share it on the screen. All right. Let's see. So this is what it looks like. Are you guys familiar with this? Kirby-Bauer disc diffusion. Who here is familiar with this one? Because I'll help you guys out if you don't. It's okay. You need not be shy with me. Because I know you guys have limited resources of learning, especially when the pandemic hit. So this is what it looks like. So it can come in packages of this one. Okay. Nicotinamine and actually the labels are actually X and V factor. It's easy for you guys to remember. All right. So it goes like it basically looks like this. So you add it on mannitol salt. Not mannitol salt agar. You use a different agar for that one. Okay, we'll talk about it later. I'll leave it to you guys to remember. Okay, so that's what it looks like. So you guys are familiar. You guys are familiar now. That's what you use for antimicrobial susceptibility testing. And even though we have automated devices, sometimes we still have to rely on our manual methodologies. Not just in microbiology, but also in other sections of the laboratory. Okay, so be mindful of these things. So you guys were prior to this lecture, you guys have to be honest with me. You have not seen what it looks like. You can be honest with me. Eliza, you have not seen it before in your life? The one that I showed you? Ay, sorry. Hindi mo pala nakita. Oo nga pala, no. Kasi naman, nandun pa pala yung ano ko. Sorry naman. Sorry na, Eliza. Okay? I'm sorry, Eliza. So, this is a... let me stop sharing the screen for a while. And let me go to share screen. Again, let's... I'll show it to you. I'll show it to you guys. Hang on a sec. So I was thinking of the live stream. So because we have the lectures in the live stream, so I was thinking you guys were looking at the live stream. Sorry. So this is what it looks like. I Google search it for you. Hang on a sec. This is what it looks like. So very cheap. I know my slideshow is very cheap, but you guys can get some sort of idea. So you guys are familiar now. Minty, nakakita ka na ba ng ganyan? Dati? Okay. What about Sabado? Nakakita ka na din ng ganyan?
Hindi pa po, sir.
Pati sa antimicrobial susceptibility testing, hindi pa din? Minty? Sabado?
Sir,
Hindi din.
pinagawa po ata kami ni Sir Mac nun.
Pero you did not do. It's okay, don't worry. I'll help you guys out. Don't worry. Next, I think on the 14th, I'm working in the laboratory. I'm still with you guys, I suppose. October 14th, I'm not sure. I'll give you guys like a short tour of what we talked about earlier, okay, in our previous discussion. So you guys don't get so you guys won't fall behind in your studies, because it's hard to study this without actual experience. Correct? Right. So I'll be showing you guys what we do in the laboratory. Is that okay ba yon? Gusto nyo ba yon? Or wag na sir, tuloy-tuloy lang ng discussion kasi ibabagsak kami ni Sir Mac. Kasi ang main goal namin ni Sir Mac, ibagsak kayo. Charot lang. Ano ba? Hindi ibagsak kayo talaga. Ang goal namin ni Sir Mac is pumasa kayo. Pero pinapahirapan. Okay? Di ba? Ganon. Bakit? Kasi para pagdating nyo sa exams, hey, ang dali-dali lang naman ang question na to. Ganon. Yung kayo pa yung magagalit dun sa examiner sa PRC. Napakadali naman ito. Mas mahirap pa yung mga pinasasagutan sa amin ni Sir Mac. Di ba? Ganon. Okay? So, yeah. NAD or nicotinamide adenine, dinucleotide, or hemine or porphyrin. So what's the difference between hemine and porphyrin? Who can give me an answer for this one? What's the difference between hemine and porphyrin? Oh, ayan na. Lumalabas na ang credit card ni Sir. Ayan na. Sige. Who can answer? Sige, who can answer? What's the difference between hemine and porphyrin? It's not related to microbiology, but it's just fun to answer because it is important in your hematology classes or when you're taking your hematology exams. What's the difference between hemine and porphyrin? Wala. Nobody? 100 Gcash on the line? Nobody? Diandra, baka gusto ulit ni Diandra ng prize. Nandiyan ba si Diandra? Diandra, are you there? Ay, wala. Wala pala si Diandra. Anyway, who can answer? What's the difference between hemine and porphyrin? I'll start the timer at 1 minute. Okay. Hemine and porphyrin. Toot. Nobody? Mark, I know you're looking at Google. I know you're Googling things. Who? What about Minty? Itaas ang bandera. No clue? What about Eliza? Hemin and porphyrin. Ayan na. I love it. Just in time before mag-stop ang timer ni sir. Sige. Who can answer? Who was that? What? I saw James Paul's hand raised earlier. Sige, James Paul.
Try lang po. Yung porphyrin po is for functions ng hemoglobin. Pero yung hemin po, pain po yung degradation product of hemoglobin.
Degradation product of hemoglobin. Yeah, that's correct. You may send me your. That is correct. So basically, they are part of the same thing. Okay, they are part of the same thing. Porphyrin is actually a precursor molecule of hemoglobin. Okay, but we use hemin here we added hemin here because hemin can be the degraded form of porphyrin or hemoglobin in general. So this one is a precursor molecule, while this one is a degradation product. So for example, I'll give you an example, guys. When you heat your blood agar, it will turn from red to brown. What's present? Porphyrin or hemin? What's present? Sige. It's hemin. Okay? It's hemin because it is the degradation product. Okay? So hemoglobin is degraded into globin chains or coagulated globin chains while the heme molecule turns to hemin. Okay? All right? You understand? Understood? Cool beans. Let's move on to the next question. What happened to my slideshow? Why is it freaking out? My slideshow is freaking out. I'm sorry. Salt-loving organisms, halophiles. Where were we? It's freaking out. So, sources of factor V, or what we call NAD, or nicotinamide adenine dinucleotide, is potato extracts, yeast extracts, and some other bacteria. Okay, so other bacteria can be the source of factor V. So please be mindful of the extracts that you have. Remember, look at the commonality between between the first two bullets: potato extracts and yeast. What are they most commonly? They're most commonly known for? They're usually carbohydrate-rich carbohydrate-rich sources. Okay, so be mindful of that one. Okay, now there are 3 organisms that you can use as factor V analogs. When I say a factor V analog, so for example, you don't have yeast extract in your laboratory, you run out of yeast extract, or you run out of bottled potato extract. You still have stock cultures of my control organisms in the laboratory. So yes, at one point in time, I relied on this one to do Haemophilus satellitism, because we wanted to confirm if the Vibrio species that I'm working with actually has factor V. Because one way to differentiate the species of the pathologic species of Haemophilus is through the demonstration of factor X and factor V. So we ran out of potato extract and yeast extract. Okay. So what I did was, I remember there's bacteria. We have stock cultures of microorganisms, control organisms, and I know that one of them was recently used and freshly planted, and it's actually Staphylococcus, right? Staphylococcus is actually one of the sources. Okay, and Neisseria can also be used, in pneumococcus, but the most common one is Staphylococcus species. Any species of Staphylococcus except for Staph albus can actually produce NAD. Okay, so I had their Staph. Actually, it was it was a MRSA strain, but it was the only Staph strain that I had at the time, and when the physicians wanted the physicians wanted to know what type of Haemophilus it was, because we wanted to have a finished report by the end of the patient's stay. So here in the Middle East, you wouldn't be able to get discharged without the final diagnosis. So they needed to write something, and part of the policy means that say you cannot say Haemophilus species. Okay, you can say that for patients who would be transferred to other hospitals, but for discharge purposes, you need to have a specific microorganism in our policy. So it's either we send out the organisms, or we detect what specific organism or the genera of the organism there is. So in cases like these, you need to be able to think fast on your feet. Should I send it? It will cost more for this to the patient, or would it be, or it would add additional headaches to the physicians in the hospital. So at that point, we decided on just planting it beside Staphylococcus, and lo and behold, the organism did what you call bacterial satellitism, when it grows near the colonies of another organism. Okay, so let me just give you a brief introduction on bacterial satellitism. By the way, hang on a sec. I'm not sure if you are familiar with bacterial satellitism, so I'll show it to you guys. So this is the staph organism that for example, the yellow ones. You guys can see it in the chat. In our lecture. So this is an example. For example, this is your staph aureus. So I'll denote it as violet because it's gram positive. Okay, all right. So I streaked a colony of staph controls. Okay, control organisms. So be mindful of the term control organisms. So they would grow probably like this in the streak. Okay, and usually it's if it's a streak, it will be like this. Imagine this is a close up view of the blood agar that you have. Okay, so because it's staph aureus, it's beta hemolytic. Okay, so everything else is clear. Everything else is clear except for this part here. Okay, so imagine this is your blood agar. Okay, so that's the streak that you did with your loop full. All right, and then, oops, what happened? Ano ba yan, sir? Pati ba naman paint, hindi mo kayang i-operate? Ano ba? Nakakainis ka. Hindi ka na tech savvy katulad namin. Ganon. Okay? So, that's your blood agar. Okay? This streak is beta hemolytic, so you guys know that it causes clear zones. All right? So, imagine this is Haemophilus, the white ones that I grow. Sorry. The white ones that I grow is Haemophilus. Platelet satellitism is similar, is like this. Yeah. A platelet satellitism tuloy. Bacterial satellitism. Where is the color? What happened to my color? So bacterial satellitism grows something like this. So the organisms will grow close, but not too close. Okay. So they wouldn't grow here, because it's the portion of the staph that grows, but it will basically just satellite near the agar, near the streak where you put it. Okay, so they'll make a demarcation line and they would satellite it. Why? Because the area around it is basically rich with NAD. Okay, so they love it there. Oh my gosh, if I'm a Haemophilus, I go, oh my gosh, there's NAD. It's rich. So I'll go near those colonies where there's NAD. I'll send my colony forming units. Okay? So the colonies will form after 24 hours, and then you're happy because you know that your organism is NAD positive. Okay? Clear? Is it clear? All right. Thank you. Thank you, Haemophilus. All right. So Neisseria species can have that. It's hard to grow them, so I'd rather not use Neisseria species. Pneumococcus, it's difficult. It's very rare that you'll see it in the laboratory, but yeah, fine. You can also find it. But the most important one that you need to remember is Staphylococcus species. So you might be asked a question: which of the following can be used as a factor V analog? So you can use Staphylococcus there. They'll give you a selection of microorganisms like right, Legionella, Listeria, Vibrio, and then Staphylococcus, and then Streptococcus pyogenes. Ha? Which one? So hindi tinuro ni Sir Manuel yon. Hoy, binago lang nila yung question. Okay? Hindi tinuro ni Sir Manuel yon. Totoo, tinuro ko yon, hindi nyo lang binasa siguro, hindi nyo lang ninamnam, or baka hindi nyo lang ina-understand. That's the reason why I want you guys to focus on how the questions we want you guys to focus on test taking skills rather than memorizing the questions. Okay, we don't want you guys to memorize the questions. We want you guys to understand how the examiner's minds work. Because sometimes they would paraphrase questions, and you guys would be flabbergasted with the questions. So they would sometimes give you a question that's extremely lengthy, like this lengthy, and it's only asking for one thing. And all you needed to do was actually look at some of the things. So one specific word there. That's your organism already. And that's the reason why I'm taking so much time in the introductions, because you've already had the specific discussions in your previous years. This is just basically a review. Okay? All right. So let's move on to the next one. Differentiate factor X and factor V on the basis of heat stability. Factor X is much more heat stable. Why? Because it's already a degraded form, right? Hemine is already degraded. All right, it's already from the what do you call this, coagulated, or the coagulated form of hemoglobin. So essentially, it's much more heat stable. Okay, so that's the only thing that you need to remember. Factor B is much more heat labile because it's a substance that is released or that is present in a particular extract. So see, it's easy to remember. Factor X is heat stable because it's derived from degraded hemoglobin, while factor B is heat labile because it hasn't been exposed to any type of extreme temperature manipulation. Okay? Now, obligate aerobes are organisms that grow only in the presence of oxygen. What enzymes are present in these type of bacteria? Sige. Organisms that are obligate aerobes are organisms that grow only in the presence of oxygen. What enzymes are present in these types of bacteria? Sige nga. Who can answer? Oxygen. I'll give you an example, a clue. Okay? One of the positive results of this test is effervescence or bubble formation. This is actually one of the, if you remember your studies in the clinical lab. Oh, somebody is already answering. Alexis.
Catalase po.
Catalase.
Test po.
Very good. Very good. Catalase test. Effervescence. Right? So the positive result is effervescence. So organisms that are obligate aerobes usually have catalase. Okay? Another one is, of course, sulfonamide dismutase. Okay? So, superperoxide, sulfonamide tuloy. Superoxide dismutase. Medyo nakakalimot si sir ng microbiology na may signs of aging, guys. Superoxide dismutase. Ang dismutase lang ang naalala ni Sir Manuel. Grabe. Dismutase lang. Superoxide dismutase. So basically, these two enzymes react. I'm not gonna delve deeper in the reaction. You guys just need to know that organisms that are obligate aerobes or require only the presence of oxygen in terms of atmospheric requirement, organisms that have catalase and superoxide dismutase. Okay? Now, examples of bacteria that are obligate aerobes. I'll give you an example of, I'll give you a mnemonic. Nagging pests must breathe. Diba? Ano yun, sir? That stands for Nocardia, Pseudomonas, Mycobacterium, okay, specifically Mycobacterium tuberculosis, and Bacillus. These are all obligate aerobes. Okay? And these are the organisms that we're going to talk about when we talk about medical bacteriology. So please don't forget this little mnemonic that I took from, I think, Microbiology for Dummies. If you see the book in National Bookstore, you'll see a caricature book of microbiology for dummies. I'm not sure if you guys are familiar. Rarely do I see my students buy other books related to academics. Mostly it's related to entertainment nowadays. Kasi ganun tayo, 'di ba? Kung ano yung interesting, yun na rin bibilhin natin. Ano bang interesting sa microbiology, 'di ba? 'Di ba Valiant? Mas maganda pang bumili ng Elle magazine, 'di ba? No?
Opo.
'Di ba? Ganon. Ba't ba ako bibili ng microbiology for dummies? Okay? Alright. So, because we're talking about oxygen requirement or the atmospheric requirements of microorganisms, we need to also talk about obligate anaerobes, meaning the absence or the absolute absence of oxygen. What are the examples of these types of organisms? Can't breathe air. O, di ba? Ano na naman yan, sir? Pinapahirapan mo kami. Hindi ko kayo pinapahirapan, pinapadali ko nga para sa inyo eh. Can't breathe air. O, di ba? Nagging pests must breathe. O, nocardia, pseudomonas, mycobacterium tuberculosis, bacillus. This one, who can answer this mnemonic? Can't breathe air. We already talked about the organisms last week. So please help me out here. What are the organisms that I'm talking about when it comes to obligate anaerobiosis? Or air, okay. Who was that? I didn't see. I'm sorry, I didn't see. Can't breathe air. O yan na, Mark. Sige, go. Ano?
Sir, dalawa lang yung naalala ko eh.
Sige lang. At
Na,
least meron kang naalala.
Actinomyces tsaka Clostridium po.
Very good. What about B? Eliza, what's B?
Bacteroids.
Bacteroides. Very good. Thank you so much.
Bacteroides.
Love it. Love it, guys. Okay? Madami ako students. Wag ka mag-alala. Hindi lang ikaw yung nagbasa na ganon. Don't worry. It doesn't matter how you pronounce it as long as, ano nga yun? You select the correct answer. Okay? It doesn't matter. I'm not going to judge you. We're not here for IELTS. We're not here for an English exam. We're here for microbiology. So don't worry. I don't really care how you pronounce it. Okay? As long as you know what the organism I'm talking about is. Okay? So Clostridium, Bacteroides, and Actinomyces. Okay? What specific organism are we talking about when it comes to Actinomyces? Actinomyces israeli. So tandaan nyo na para hindi nyo makalimutan. Okay? Alright. So, let's talk about the term given to an organism that requires a lesser amount of oxygen. It's actually known as microaerophile. Okay? So a microaerophile. I'll show you guys how we do microaerophilic environment. Microaerophilic setup. Okay, so a microaerophilic setup is something like this. It's like this one. I remember I Google searched it for my previous class before, and it's still in my history. So hopefully you guys can see it. Can you guys see it? Mark?
Hindi pa po. Hindi pa po.
All right. So sorry. Oh, because I wasn't sharing my screen yet. What about if I move it like this? Can you guys see it if I move it here?
Yes po.
All right. So I'll move it here like that na lang. So look at the left side, okay? So for microaerophilic environments, you need this particular setup. Okay? So this is a cylinder. Okay? So you have the cylinder here. You have the borosilicate cylinder, actually. And then you have a lid with a gasket. What you put inside is an envelope containing sodium bicarbonate and sodium borohydride. Borohydride. And what it does is it consumes a certain amount of It consumes. I think this is a similar setup for anaerobic setups. But just for your information, um, what happens in microaerophilic environments is that you want to have at least 3% to 5% oxygen saturation within that. Sorry, 5 to 6%. Sorry. So, elderly na talaga si Sir Manuel. At least nakuha ko yung 3 to 5. Actually, 3 to 5 sa laboratory namin eh. Kaya yun pala yung naalala ko. So, 5 to 6 pala siya ideally in the books. Okay? So, you need 5 to 6% oxygen. All right? Now, what happens is you put them in a gas pack. This is known as the whole setup here is a gas pack. Your plates or your petri dishes are placed inside, and besides them, you're going to put the microaerophilic gas packs. So what it does is during the duration of the incubation, the whole setup loses a certain amount of oxygen, okay, leaving only at least about 5 to 6%. That is, how do they know this? They've done controls on it, okay, before sending it out to us. It's already pre-packaged. So it's like Nor cubes. Diba? You're putting something in your soup with a certain level of umami that has been tested by several people. So it's like that. Okay? So imagine it like a pre-packaged version. It's like a sachet. It's like you put it in there. It's already prepared for you. All you need to do is incubate it. Let's see what's inside. So it's like a choose your own adventure kind of thing. All right. So that's basically what a gas pack is, and it's the same setup for anaerobic systems. So again, you have a gas pack. What's the difference is that it has palladium pellets. It removes the oxygen. It's like EDTA for gas. What does EDTA do? It chelates the calcium to prevent coagulation. So in this case, palladium pellets chelates the oxygen from the whole setup so that the organisms that's anaerobic or microaerophilic can grow and thrive in that situation. So, okay? So hopefully you guys had an understanding. Okay ba? We can move on. Is it okay? Can we move on? Mark? You
Yes
speak
po.
on behalf of the class because you are my moderator. I know it's an
Yes po.
unfortunate. I know it's an unfortunate thing, but don't worry. Sir Marco rewards people who have the tenacity to do both the moderating activities as well as studying. So don't worry, you will be rewarded for your efforts. Okay, now gram-negative curved rod, that is microaerophilic. We're talking about a medically important bacteria, okay? Which gram-negative curved rod, so meaning to say, it's a gram-negative bacilli. So why did I, grabe, bakit kailangan mo pa magpaltan-paltan, sir? Bakit kailangan mo kami pahirapan? Kasi ganun ang ginagawa ng mga examiner there. They try to paraphrase the questions. So, gram-negative curved rod, meaning it's a bacillus or a bacilli. Okay, the shape of it is a bacilli. That is microaerophilic. Choose that one that is medically important. This is a very important question. You need to mark number 82, write it in your papers because this is important. Okay? They would ask you a question, and it starts with a case study, and then you're going to see at the bottom, or the organism grows at a microaerophilic setup of 5 to 6% oxygen saturation. Oh. Yeah, that's the reason why I'm asking these questions. Okay, so who can give me a microaerophilic organism that's commonly grown in the laboratory as a pathogen? It's a causative agent of um ulcerations. Yeah, this family of microorganism grows specifically to cause ulcerations in the stomach. Okay. What else is there? It's capable of causing gastrointestinal ulcerations as well. Specifically, as I mentioned before, it's in the stomach. Another one is on the lining of the ileum. Yeah. Who is that? Valiant or Minty?
Helicobacter pylori.
Okay, that is actually correct. but they belong to the species today. Right now, they belong to the species of Campylobacter. But yeah, that is correct. Helicobacter pylori was sorry, yeah. Oh, I'm actually the one that is incorrect. It's not Campylobacter anymore. It's Helicobacter. Okay? So please change this one to Helicobacter. I'll edit this one later on, but please remember to write Helicobacter pylori. Okay? So Helicobacter, but they still belong to the family Campylobacter. It just so happens that the genetic sequences makes them different from Campylobacter species. Okay, so they're still under the same umbrella, okay, of microaerophiles. What's the other organism under the Campylobacter species? We have Campi agar. Okay. By the way, we have Campi agar in the laboratory. Ha? What's Campi agar? Campi agar is a blood agar with a lot of antibiotics that prevents the growth of other GIT related organisms. Okay, and it only selectively grows Campylobacter species. Campylobacter jejuni, right? Or jejuni, depending on how you pronounce it. But again, if we're talking about it, if you're talking about these things, you have to include Helicobacter as well. Okay? Alright? Sir, paano namin ma-differentiate sa laboratory kung mag-grow si organism? Kung Campylobacter siya or Helicobacter. It's very easy. We have H. pylori kits. It's like a pregnancy test. Okay? I'm not sure if you've discussed this in analysis of urine and bodily fluids, but one of the routine tests that they do in well-off countries, I think it's not common in the Philippines to have H. pylori infections. Um, I think I could remember a handful of cases where H. pylori was the organism to be considered. I think 5 or 3 organisms throughout my whole 2 years of working there in the Philippines. I only experienced like a handful of tests that require H. pylori. But here in the Middle East, and I think other people from Western countries like the UK and the United States, it's much more prevalent there because a lot of people they eat with their bare hands. Okay, so west of Southeast Asia, there's a lot of infections of H. pylori. So how do they do it? It's basically like pregnancy test, but you have to make a stool suspension of a patient's of a patient sample and then drop it on a kit similar to drop that suspension on a kit similar to a pregnancy test kit, and that's basically it. That's how you can differentiate it. It's more or less chromatography, because the basis of your pregnancy test kits are color chromatography. So you'll see a line. If it's one line, it means that it means to say it's positive, and if there's only if there's two lines, it means to say it's positive. If it's one line, it's negative. Okay. Indeterminate if there's no lines, meaning your controls are not okay. Okay? Okay. Now, let's move on to capnophilic organisms. So what are capnophilic organisms? What do they require? They require CO2. Okay? CO2. Okay? So the most commonly used techniques in the laboratory to create a capnophilic environment or a capnophilic atmosphere for your microorganisms is candle jars. The usage of candle jars. Again, I'll Google search it for you guys because alam nyo naman guys, I love you guys so much. So I'll show you guys. Have you guys seen a candle jar setup before? You can be honest, don't worry. If you don't know. What about Arnie? Have you seen a capnophilic setup before? Sorry, a candle jar setup.
Nothing, sir.
Okay. What about Clarice? Have you seen a candle jar setup before?
Pictures lang po, pero yung actual, di pa po.
Hindi pa din. Okay, so what did you see in your picture, Clarice? Sabi mo nakakita ka eh. What did you see? Nakamute ka te. What did you see in your capnophilic setups? Okay, so wala siyang idea. So let's candle jar. Okay, I'll show this to you guys. Again, it's Google searched. I don't take any what do you call this? I don't take any credit for this one. So a traditional candle jar setup is like this. Huh? What happened? What happened to me? Can you guys see? Not sure. I'm not sure if you guys can see.
Yes, sir.
All right. So a traditional candle jar setup is like this. You basically put it in a glass jar, right? And you have a lid, you have a glass jar, you have tube media, you also have petri dishes, and on top of it you put a candle. You light a candle on it. What's the principle? Who here knows the principle of a candle jar? Who can explain? Nigel, explain to me the principle of a candle jar setup.
Yung candles po, yung oxygen dun sa jar, mako-consume yung candle. So,
Very good.
better.
I love it. I love it. O, di ba? Ganon. Ganon ang mga sagutan. Witty na pretty pa. I love it. I love it. Love it. Ganon. Okay? Alright, so what is the preferred percentage of candle jar setups? Okay? So what is the preferred percentage? Ito na naman tayo sa percentages, sir. Kailangan ba naming alalahanin yan? Yes, it's important. Okay, it's important to know this. Okay? Okay, kung mag-aaral na kayo ito, doon yun na ha. Alright? Okay, so how many percent of CO2 do we need in our candle jar setups? Eliza.
5 to 10% po.
Very good. Very good, very good, very good. Very good. Okay, now, let's talk about capnophilic organisms. Alright? What do you mean capnophilic? Sir, ano ba yung mga capnophilic na yan, sir? These belong under the Hasek group, also known as your fastidious organisms or fastidious bacilli. There is a specific discussion on this after we discuss other microorganisms. I usually discuss this at the very last part of it, because at the very last part of our lectures, because I don't want to confuse you guys with general knowledge that needs to be understood. So basically, HASEC stands for Haemophilus, Actinobacillus, Cardiobacillus, Echinella, and Kingella. Okay? HASEC. Alright? So please remember your HASEC group. They are usually the ones that require CO2 in their systems. Okay? CO2 in their system. So that's like Haemophilus, Actinobacillus, not Actinomyces, Actinobacillus, Cardiobacterium or Cardiobacteria, Echinella, and Kingella. What about examples of capnophilic cocci, sir? Who here can give me an example of capnophilic cocci? I think the boys can help me out here. Diba? The boys. Excluding Minty. Excluding Minty. I don't know what you do, Minty, but whatever it is, I'm going to exclude you na because I am categorizing you as one of us. Okay? Yes. Good darn. So, boys, I think the boys can answer this one because this one is easy for the boys. Yung mga tao na may tulo, ano yung napapatubo natin doon? Pag may tulo ka, Mark, anong sakit mo?
Gonorrhea po.
Oh, ano yung family nun?
Nakalimutan ko.
Hoy! Kakaloka kayo. Boys muna, girls. Iwanan muna natin. Hindi ko muna kayo tatanungin. Baka
Sir,
sabihin kasi ng mga
sir,
boys.
Neisseria. Gonorrhea.
Oh, nakakainis ka, Mark, ha? You have to put in an effort in your studies. Hindi pwedeng may plus 10 ka lagi. Hindi pwedeng
Yes,
mag-rely. Don't rely on that plus 10.
Yes, po.
parang si Valiant. Don't rely on that body. Stop relying on that body. Hindi ka hanggang doon ka lang hanggang at a certain point your body, Valiant, will not take you to to stardom, okay? You have to show brains as well, ha? Okay? 'Di ba, Valiant? Kailangan hindi pwedeng pretty lang, Mark, ha? Kailangan smart din, okay? All right. Okay, next. Let's move on. So I love that you guys are much more active than my previous classes. I suppose you guys are much more experienced in asynchronous learning. So yeah. Okay, so these are your Hasec species: Haemophilus, Actinobacillus, Cardiobacterium, Echinella, and Kingella. Okay, you have Capnophilic cocci, Neisseria. Okay. Next, aerobic cultures require what percentage of oxygen ideally? Well, to tell you frankly, you just need 21% oxygen. Okay? Not really 100% because 100% oxygen can be detrimental to microorganisms. Yung konti lang. I think in the Philippines, uso lagi, I think hindi na uso, matagal na yung pauso ng mga Pilipino, yung sakto lang. Ganon. Yung sakto lang, sir. Hindi yung 100 exactly. Sometimes kasi kapag hindi sakto lang, 'di ba? How are you today? How are you today, Nigel? Sakto lang. Ganon, 'di ba? How are you today, Clarice? Next time pag tinanong ko kayo, Clarice, Nigel, pag tinanong ko kayo, how are you today? Sakto lang, sir. Ganon. Kasi hindi ka sobra ng oxygen, 'di ba? Nakakaloka kayo. Nakakaloka kayo. Natutuwa ako pag natatawa kayo sa mga jokes ko. Kasi ang corny ko talaga. Ang corny ko talaga, guys. Okay? So, how much would it ideally require for an anaerobic culture? So, how much H or hydrogen do we need? How much CO2 do we need? How much nitrogen do we need? Bakit naglalagay ka pa ng 2, sir? Yun lang naman yun. Kasi ayoko makalimutan yung chemistry basics nyo, ha? Ayoko. Baka mamaya sabihin nyo, microbiology naman tayo eh. Baka mamaya. No, I'm approaching every lecture with you guys as a holistic approach. So ayoko kayo na nagfo-fall behind. Pagka mga students ko, ayoko. Kailangan smart na pretty pa. Katulad ni Nigel, o 'di ba? Nigel, ha? Smart and pretty, ha? Hindi pwedeng smart lang. Hindi kailangan nag-wake up ka pa rin kahit na perfect mo ang kahit na perfect ang exams, okay? Magpa-contour ka kay Valiant, feeling ko alam niya kung paano. Magpa-contour ka diyan, okay? Alright. So how many percentage? Sir, kailangan pa talaga naming alalahanin? Yes. Huwag niyo kakalimutan na 5 to 10% lang lagi ang pinagdadaanan ng mga mikrobyo natin. 5 to 10% H2, 5 to 10% CO2, 80 to 90% 80 to 90% nitrogen. Okay? So yan. Madali lang naman ang computation. Okay? Mga 20 ang babawas nyo. Then, okay. Alright. 100%, sir. Bakit walang oxygen, sir? Kasi nga anaerobic. Okay? Okay, magulo ha. Anaerobic nga eh. Ba't maglalagay ka ng oxygen dyan? Sasakalin ko kayo. Okay? Pag merong, paano sir kung merong 1%? Di microaerophilic na yon. Walang etchosera dito ha. Baka mamaya etchosin nyo ako ha. Sir, bakit ganun? Hindi tumutubo yung microorganisms ko. Paano kasi hindi gumana yung anaerobic system mo. Okay? Meron ako dating intern, naiinis siya sa akin. Sir, naiinis ako. Meron ako 1% oxygen. Sabi ko, gaga, nakabukas kasi yung hindi tightly sealed yung anaerobic, yung gas pack niya. Kailangan tightly sealed yun. Kailangan yun ng boys. Kaya nga mas gusto ko kapag kami mga interns ako, kailangan may lalaki na isa. Kasi kailangan siya yung magagano ng gas pack. Kasi mahirap talaga siyang isara. Lalo na kapag anaerobic cultures. Kailangan talaga pag may intern kami. Kasi yung mga girls, gaganyan-ganyan lang. Tapos problema, sir, parang hindi nag-change yung color ng color indicator. Paano? Bruha, hindi mo kasi kailangan may muscle dapat. Pagka ganon. Okay? Hindi pwedeng pa-inday-inday pag nag-internship kayo. Mag-internship na ba kayo or nag-intern na kayo? Hindi ko na alam kasi ngayon curriculum eh. Can you enlighten me, Mark, on what's happening in the education of medtechs in the Philippines?
Virtual internship po for 6 months. Tapos, nasa hospital po yung second 6 months.
Nakakasad. So, the day in the life of Sir lang, ganon. Bali, parang ganon lang.
Yes po.
Gosh! Oh my gosh! I feel so bad for you guys. Kasi hindi nyo man lang ma-experience yung yung merong internship syndrome. Nakahabag ako sa inyo. Wala man lang yung merong magjowa, papasok lang sabay sa ospital. Tapos magbe-break sa ospital, di ba? Hindi ako makakalandi sa internship. Gaga, huwag ka maglalandi sa internship. Ikaw na nga lang ang future ng mga baklang medtech sa Pilipinas tapos maglalandi ka pa. Paano kung na-fall ka tapos hindi mo na kaya, hindi mo kinaya na heartbroken ka tapos nagbagsak-bagsak yung mga subjects mo. Bruha.
Wala yung magagawa, sir. Ako bahala dyan.
Ay nako, I love it. May something
Nandiyan,
to. Pero,
nandiyan lang, nandyan lang siya sa kabilang kanto, kaya pwede kong puntahan.
okay. Akala ko may something kayong dalawa. Anyway, tapos pala Valiant ha. Pababantayan kita kay gusto ko ikaw ang future ng mga medtech ng mga bakla sa Pilipinas. Gusto ko isa ka sa magiging kasi when I was there, I was actively when I was there in the Philippines, I was actively promoting HIV awareness. I wanted because in the Philippines it's really the what do you call this, the state of our HIV infection rate is really sad. It's really saddening, right? Like one container would have 99 people would be positive and only one would be negative for confirmatory HIV testing. So talagang ayoko talaga pagka as much as possible kapag binibigyan ako ng time mag-lecture, lagi kong ini-emphasize, lalo na kapag microbiology, lagi kong ini-emphasize yung HIV awareness. So I want you to take the throne for me for 2023, Valiant, okay? For 2023, you will be my HIV ambassador. Yung hindi ikaw yung magkakalat ng HIV, gaga ha. Huwag kang lalandi. Bakla ka ng taon, huwag kang lalandi ha. Okay? I want you to be the person every year I would select somebody who would take the reins, okay? So at least try your best if you have younger people, okay? If you have younger people, maybe first year students who may not be included in medtech, who may not be enrolled in the College of Medtech. Yeah, ganon. HIV counselor. Very good. Actually, they're high-paying jobs, to be honest with you. Pero ang hirap kasi dyan, nakakabwisit kasi dyan sa Pilipinas, kailangan HIV proficient ka pa. Magka-counsel ka lang naman ng student. Ewan ko dyan, nabubwisit ako dyan sa Pilipinas, yung sistema natin dyan. Di ba? Kailangan HIV proficient pa para sabi ka lang naman, ganito yung mga kailangan mong gawin. Hindi ka na pwedeng magganyan-ganyan kasi na-infect ka. Anong kailangan? Paano magdidip? Yun lang naman gagawin ninyo doon sa sa HIV proficiency testing. Wag ganyan lang kayo ng HIV strips tsaka yung HIV comb. Yung pang-test ng tapos kakain lang kayo. Yun ang binayaran ng mga companies ninyo na 10,000. 10,000 yun. Tapos ma-stuck ka pa sa Pilipinas dahil hindi ka makaalis. 'Di ba? So, yun. Anyway, nawawala na tayo sa discussion natin. So, yun. Basta babantayan ni gusto ko apart from your dadagdagan ko na naman ng another thing ang responsibilities ni Mark, bantayan si Minty, ang innocence ni Minty.
Kaso sir, hindi na po yan innocent.
Ay, girl, alam ko naman din, pero at least kahit papano we should preventive measures should be taken. Okay? Preventive measures should be taken. All right. Now, let's move on to slide number 87. We're getting off track again. You guys are distracting me. Minty ha, stop it. I know what you're doing. You're distracting me. Okay? I know what you guys are doing. Charot. Charing lang. All right. Anaerobic jars, containers, bags prevent the accumulation of oxygen due to what catalyst? So as I mentioned before, these are metals. Specifically a metal. It's palladium pellets. Okay? And the indicator that we can use is either resazurin or methylene blue. Okay? Resazurin or methylene blue. So what's the importance of these two color indicators? They indicate the presence of oxygen. Okay? So I'll give you a question. Indicate the reaction of anaerobiosis. What do you mean by anaerobiosis? Okay? So they are metabolizing something. So when you say biosis, it has some, hindi bayot. Okay, hindi bayot, Valiant. Kakaloka ka, Valiant. Tumatawa ka na naman, ha? Valiant, ha? Wag ganon. Nakakaloka ka talaga, Minty. Anaerobiosis means walang kinalaman sa sexuality yan, ha, guys. Sampalin ko kayo. Alright? Tumatawa si ano. Nakikita ko gumaganyan yung gumaganyan si Minty, eh. Wag ganon. Okay? So, anaerobiosis. So, anaerobiosis is basically basic biological functions in the presence of, or without the presence of oxygen. Okay? Aerobiosis with the presence of oxygen. Anaerobiosis, you're just removing oxygen out of the equation. Alright? So, using methylene blue, what would be the positive, what would be the negative? Who can answer this one? Positive. What would be the color indicator? What would be the reaction of your color indicator if there is negative anaerobiosis, meaning there is oxygen present? Nakakalito. Nakakalito. Nalito kayo. Tagalogin natin para madalian kayo. Kasi babalik-tarin ko yan sa exam. Gusto ko yung nalilito kayo. Kaya kailangan ngayon pa lang naintindihan nyo na. Anaerobiosis. Negative anaerobiosis, meaning there is oxygen. Diba? Negative anaerobiosis nga eh. Anaerobiosis by itself means that there is no oxygen. So a negative reaction means there is oxygen. So what would be the color of methylene blue? Is it blue or colorless? O yan na, binigyan ko na kayo ng choices. Sige, type it in the chat. Type it in the chat, guys. Gusto ko makita yung Gusto ko makita lahat ng tao. Blue, according to Valiant. Sige. What about Nigel? Sige lang, there is no wrong answer. Don't worry. Hindi pa tayo dito. Hindi pa to graded. Huwag kayong mag-alala. Okay. Meaning may oxygen, ha? Meaning may oxygen yan. Negative anaerobiosis nga eh. Sige. Ay, taray, kumpleto talaga, oh. Ayaw magpatalbog kay Valiant nito ni McKay. Talaga ayaw magpatalbog. Work. Ganun ako. Work. Ganun. Work. Alright, somebody said negative is colorless. Okay. Somebody said negative is colorless. Okay. Alright. Okay. Let's see. Correct answers are as follows. Negative, meaning without oxygen, it's white. Positive, meaning with oxygen, it's blue. So there's only a handful of people who answered correctly. So it's Johaili, Maria Cristina, and Amanda Feliz. Thank you guys so much for answering. Don't worry, hindi naman kayo graded. Most of the people answered. So, kailangan tandaan nyo na yon, okay? Meaning walang oxygen. Bakit? Saan ba nagre-rely ang reaction ni methylene blue? Sa presence ng oxygen. Kaya nga siya nagiging methylene blue dahil nag-oxidize siya. Kung walang oxygen, colorless siya. Gets? Get? Ow! Gadarn. Okay? Okay ba? Nagets nyo? Alright. Alright, let's talk about psychrophilic organisms. What do you mean by psychrophilia or psychrophilic organisms? Sige, general lang tayo. Psychrophilic organisms are organisms that thrive in what temperature? Psychrophilic. Cold. Ayan na nga, cold enrichment. Diba? Ideally at room temperature. Ayan na nga, cold. Okay? Ideally at what temperature? At room temp. Psychrophilic organisms, to be specific, are at room temperature. Actually, cold enrichment media requires negative 0. 0 degrees pala. Examples of organisms that thrive in psychrophilic environments are Listeria monocytogenes and Yersinia enterocolitica. So be mindful of this one. Okay? Yersinia enterocolitica and Listeria monocytogenes. Sometimes, pag nakalimutan ninyo yung lahat ng detalye ni Listeria. For example, catalase positive, catalase negative, blah, blah, blah. Produces sulfur, blah, 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 blah. Produces streamers. Stalactite streamers. Yung mga ganon. Nakalimutan nyo kunyari yon. Then, nakalagay doon, cold enrichment media. Adjusted at a temperature of 0 degrees Celsius. Alam ko na yan. It's either Yersinia enterocolitica or Listeria monocytogenes. Ngayon, differentiate mo na lang siya kung paano. Ano ba difference nilang dalawa? Ano ba what's the main difference between Yersinia and Listeria? Sige, balik tayo sa microbiology basics. Sige, Listeria. Sige guys. Anong pinagkaiba nila? Pareha silang bacillus. Pero anong pinagkaiba nila? What's the first thing you do when you when you cultivate an organism rather, or a unknown bacterial suspension? You do what? Gram stain. You forgot na, guys. The main importance of the main importance of the importance of gram stain should not be underestimated. Kasi lahat ng what do you call this? Manual biochemical algorithms that you're going to learn in this lecture of mine will always start with gram stain. So lagi kayo mag-rely sa gram stain. Sino ang gram positive, sino ang gram negative? Sige. Nako, nakalimutan na kaagad. Sige, magtatawag ako. Sino ba ang nasa chat? Sino ba ang nasa ano? May nag-raise ka kagad ng ano. May nag-raise na kagad ng hand. So, sunwerte yung hindi ko natawag. Muntik ko nang tawagin si Nigel, sige. Muntik ko nang tawagin si Randolph sa Sugitan. Muntik ka na. Swerte ka dahil nandiyan si Nigel. Alright, Nigel. What's the difference between these two organisms?
Si Listeria po ay gram positive.
Okay.
Si Yersinia ay gram neg.
Very good. Bakit bakit merong hesitation? Why is there immense hesitation? There was such a long pause. Nakakatuwa ka. There was such a long pause. Siyempre, gram positive yung isa. Siyempre, gram negative yung isa. Ikaw talaga, girl. Ikaw ha. Niloloko mo ako. Alam nga, gram ghost? Ano sabihin mo, gram ghost? Gram neutral siguro, sir? Ano ka ba? Ikaw talaga. Nakakatuwa ka ni Jill. Natawa kasi. There was like a 5-second pause. And then may question mark pa sa dulo. Yung talagang hindi siya sure. Nakakatuwa ka. Alright. So, yan. So, under ba siya ng anong family? Enterobacteriaceae family. So, di ba parang, whilst we're discussing, I'm giving you overviews of what organisms you might expect in our discussions. Para hindi na kayo mahirapan, di ba? Di ba? Ganon mag-intro si Sir isang araw or dalawang araw. Alright, now let's talk about mesophilic organisms. So, mesophilic organisms, they thrive under room temperature. Okay? So, be mindful, psychrophilic temperatures are less than, 10 degrees. And then mesophilic meaning the what we have as atmospheric temperature is basically at around 30 to 40 degrees Celsius. And then most pathologic organisms grow at what temperature range? Most pathologic. I'm not saying all pathologic organisms, most. Okay? So, who here can answer the last one? Most pathologic organisms grow at what temperature range? Sige. Pag nagkakasakit ka, ano bang temperature mo? Maria?
30 to 37 po.
Okay, good. Good. Body temperature. But that's okay. 30 to 37. Body temperature, okay? Medyo nilalamig, nag-chills. Pwede din 'yon kasi nga the body's normal response to any microbial invasion is to increase temperature by chilling. Okay? So yeah, you're correct. 30 to 37 degrees Celsius. So temperature of most pathologic microorganisms. So when I say temperature of most pathologic organisms, hindi yung temperature nila ha. Yung temperature na requirement nila. Walang pilosopo ha. Valiant ha. Wag kang pilosopo ha. Diba? Sir, lalagyan ko ng thermometer yung colonies. Wag ganon ha. Pag nandun kayo sa laboratory, sasampalin ko talaga kayo. Pag mga ganyan yung mga ganyan na mga tanungan. Gagawin ko talagang my life's purpose talaga kapag naging intern. Pag naging intern ko tapos ganong klaseng pampipiloso po sa akin, gagawin ko my life's mission na hindi ito papasa ng NEDTech. Pagka gano'n na mga tanungan. Okay? So next, optimum pH. So we're now moving from temperature to pH. Optimum pH. Optimum pH is at around 6.5 to 7.5. Okay? 6.5 to 7.5. Now, let's talk about Lactobacillus acidophilus. Okay? It's an organism that grows best at what temperature? The only organism that you're going to talk about that thrives on extremely acidic environments, it's at a pH of 3. Another name for Lactobacillus acidophilus, it's the only time and the last time that we're going to talk about Lactobacillus acidophilus, by the way. All right? Lactobacillus' other name is Doderline bacilli, and the recommended media to grow Lactobacillus acidophilus is you would be so surprised because it would be at a TJA. What's TJA, sir? TJA, tender juicy. Ayan na naman tayo sa mga questions na nakakaloka, ha. TJA, hindi tender juicy agar, hindi yun gawa ng tender juicy or ng Pure Foods, ha. Walang ganon, ha. Walang naloloko dito. Ikaw, Mark, ha, tumatawa ka, feeling ko yun yung hula mo, TJA. Hindi. O, sino makakasabi ko ano yung recommended media for growing Lactobacillus acidophilus? Because it has a pH of 3. Sige. What's TJA? Matatawa kayo kung anong ibig sabihin ng TJA. Direct na? Or gusto ng GCash? Hindi nyo yan makikita sa Google, promise. Libro yan. Kailangan nyo magbasa ng libro ng Microbiology. wala. Sige, Eliza.
Hula lang, sir, ah.
Oh.
Ano po? Tomato juice.
Very good! Very good, Ayan! I love it! Minty, isang palakpak nga ng mga drag queen. Mali! Yung palakpak natin mga drag queen. Ganyan. Bruha, hindi ganyan. Nakakabwisit ka talaga. Kailangan mo ng orientation sa pagiging bakla. Ganon, oh. Ganyan lang. Oh. Yes, queen. Yes. Tomato juice agar. Alright? So, bakit ba? Ano bang importance ng tomato juice agar na yan? Kasi yan yung ginagamit nating controls. Actually, guys, I'll give you guys an insight of this one. Alright? Tomato juice agar is used as a media of choice for people who are developing feminine wash. Okay? So they use it as a what do you call this? Control. Because why? They want to know how strong or how potent a particular feminine wash is. Bakit? Ano ba yung Lactobacillus acidophilus? Normal flora ng vagina yan. Okay? Normal flora siya ng vagina. Walang kabastusan. No malis, ha. Ikaw talaga, si Minty talaga nung narinig lang yung word na vagina, ew! Ganun ka agad. Ay nako. Nag-aaral tayo ng science dito, ha? Huwag ganyan. Science yung inaaral natin dito. So, yun yung ginagamit kasi nila. So, si pH care, yan yung mga ganyan. Yan yung ginagamit nilang to test how potent or how strong a particular one is. By, what do we want in the vaginal tract? Do you want to I'll give you guys a scenario. Do we need to have a lot of Lactobacillus or Doderline bacilli or D bacilli na lang? L acidophilus? Or no L acidophilus? Sige. Tama ba spelling ko? Sige. Who can answer the question? We need we're studying medical microbiology, so I think it's important. It requires a bit of our attention. Sige. When you for example, you're a scientist. Nigel is a scientist working as one of the quality control personnel in the pH care laboratories. And they just developed a formula for a new feminine wash? Ano ba yan, sir? Ano ba mga klaseng tanong yan? Science-related siya. Huwag kayong etchusera dyan. Okay, tinanong ko yan actually doon sa mga students ko last time. Hindi nila nasagot. Malay nyo, itanong ko ulit sa exam. Okay. Kailangan ba merong lactobacillus acidophilus sa vagina or wala? Kailangan nating i-eradicate? Mark, sige. Being a specialist on the vaginal tract, sige.
Sir, yes po, a lot po kailangan kasi katulad ng sinabi nyo, Lactobacillus acidophilus is a normal flora in the vaginal wall.
Very good.
So kung wala, edi masisira po yung normal flora ng vaginal wall.
Very good. Kaya nga hindi pwedeng gumamit ng bareta, 'di ba, sa PH care. 'Di ba? Hindi pwedeng gumamit ng bareta. 'Di ba? Ganon yung ideation doon. That's the notion that we want to instill on you guys. The normal flora is important, okay? In the skin also, we have a lot of normal flora there. And what's the main purpose ba ng what's the main purpose of normal flora? Kasi baka mamaya magkaroon kayo ng false notion na kailangan nating eradicate lahat ng mikrobyo sa katawan ng tao. What's the importance of lactobacillus tuloy, of normal flora? Sige. Who can give me an example? Okay, ayan na naman talaga. As a specialist of the genito-vaginal tract, all right? Again, Mark Segi.
Sir, one is added protection po.
Okay, very good. Protection by what? By what? By means of what? Ayan, naku
I
pahirapan.
mean, ayun lang, sir.
Ayan na sinasabi. By competing with pathologic organisms. Diba? Sometimes, they are actually the ones who produce metabolites to destroy the other organisms. So, for example, I'll give you guys an example. Normal flora of the large intestine is Escherichia coli. If the normal flora was overcome by antibiotic susceptibility but by continuous ingestion of antimicrobial drugs, there would be a thriving population of Clostridium Clostridium perfringens Clostridium difficile. This is a bad thing. Okay? It's a bad thing. Okay, why? We don't have a lot. Normally, we have Clostridium difficile, but because we have Escherichia coli to compete with Clostridium difficile, that's the time that we maintain hindi homosexual. Bruha ka talaga, Valiant. Nakikita ko na naman you're smirking. Homeostasis. Okay, that's the time we're maintaining homeostasis. Okay, all right. So you need to have a balance of both normal floras. If one normal flora is inhabiting you or invading you too much, like in cases of Escherichia coli, that could be a bad thing. So each of them competes with each other to have a harmonious relationship with one another, thus producing an outcome of a person becoming healthy. If one of the organisms become much more numerous than the other, that results to pathogenesis. So be mindful of those kinds of things when we talk about these things in microbiology. We don't want to remove one specific population of organisms. Sabi, alsin na lang natin lahat ng microorganism. Mga walang hiya. Pag inubos niyo ang mga mikrobyo, kayo naman ang susunod. Kasi why? Genetically speaking, the reason why we have specialized cells for protection is because we evolved as humans from continuous infections. Okay? If you look at the bone marrow of if you carbon dated the bone marrow of animals from way back of monkeys, okay, from way back in a million years ago, they didn't have specialized cells for protection. Kaya nga mga itsura ng mga organelles natin, they resemble bacterial structures. Mitochondria looks like a bacilli, right? The mitochondria looks like a bacilli because it evolved we evolved from that one. So the notion that we have to remove all microorganisms to have a healthy lifestyle is wrong. You have to have bacteria. Kaya nga mas gusto ko pa ang mga bata naglalaro sa may kalsada. Except lang yung may ulan tapos basa, baka magkaroon naman ng parasite 'yon. Pero mas gusto ko pa yung na-expose sila kasi the immune system becomes they become much more competitive, or they become much more competent. A person develops a certain level of competitive a person becomes develops a certain level of immunocompetence. So hindi pwedeng ubusin natin yung mikrobyo, okay? All right, so Dodderline bacilli, best grown in tomato juice agar. Thank you so much. Now, Vibrio species grow at an alkaline range of 8 to 10. Okay? Yan yung gumagamit ka na ng borax. All right? Oh, ayan na naman, may tumawa na naman dun sa chat. Oh, may tumawa na naman dun sa may video call. Nakita ko may tumawa. Hindi po hindi po bastos yung sinabi ko, borax. Borax lang ang sinabi ko, okay? B-O-R-A-X, okay? Huwag niyo papalitan ng T. Hindi ko na sasabihin kung ano yung ano. Okay? Walang ganon. Alright? May mga ngumingisi dun sa chat. Okay? The most commonly used alkaline broth for Vibrio. Okay? Who can answer this one? Who's that? Valiant. Or Minty.
Alkaline peptone water.
Alright. APW. Thank you so much. Alright. Very good. Alkaline peptone water. Why? Because it's the only organism that will grow there. Other organisms will not grow there. Because what the pH is at 8 to 10. Okay? The pH is at 8 to 10. Right? So Vibrio. Next. Enumerate the growth cycle of bacteria. So we're talking about bacterial physiology. So we talked about bacterial basics last time. We're going to delve deep into the bacterial physiology now. So there's four phases of bacterial physiology. Okay? Bacterial growth cycle. We start with the lag phase, where there is little to no multiplication. Then there's the log phase, the growth at a maximum rate. And then we have the plateau phase, or the stationary phase, where there is cessation of growth. The last phase is, of course, decline. Growth decreases, viability count decreases as well. Okay? What do you mean by the viability count? This is when the number of organisms and metabolites are too much, that the nutrients as well as the colonial space is insufficient for proper bacterial growth. Okay? So doon makikita niyo yung mga graph na yan. Log phase will start as a straight line, and then exponential phase, and then there's a plateau phase. And then when the organism has too many metabolites, there's the death of microorganisms far encompasses the growth rate, or the logarithmic phase. It will decrease. Okay? So clear? So ano lang siya? Bali, it's a straight line, it will make a plateau, and then it will go down. Okay? So that's what happens in the incubation period. Okay? That's the reason why we only incubate at a maximum of 48 hours. After reading it initially after 24 hours, we put it back in the incubation chamber, or your incubators, okay? And then we read it the next day. Why? If it's more than 3 days, you discard the sample because it's no longer viable. There's too many metabolites. There's too many metabolites that it can contaminate the testing proceedings as well. It could be too acidic, it could be too alkaline, depending on the metabolites that the microorganisms produce. So I'll give you an example. When you run certain bacterial suspensions in a machine, kasi, okay? So gumagamit kami ng Vitek. I'm not sure if you guys are familiar with Vitek systems. It's an automated ID and sensitivity analyzer for microorganisms. We also have Bruker. I'll tell you guys about Malditov at the end of this lecture before we move on to parasitology. Okay? But for Vitek systems, how they do this is there's an automated system. And one of the most important things that the field service engineers taught us, or they're biomedical engineers, by the way, they taught us, is that if you use a 4-day-old organism, it will be misread as another organism. That's the reason why they always put in the instruction manuals, kindly use a 2-day-old microorganism, bacterial suspension. Why? Because if there is if you're analyzing colonies that are on the death or decline phase, the viability of your results become compromised. Okay? They become erroneous. Okay? So be mindful of these things. So I will maybe I'll ask a question. The plates were read 3 days prior to this one. Which of the following would most likely be a cause of error? 'Di ba? Yan na, diyan ako magtatayo. Yan ang dapat ng mga kernels of information na dapat sinusulat nyo, pero wala akong nakikitang nagsusulat. Lagot kayo sa akin. Yan yung mga kernels of information na kailangang inaalala ninyo. Kasi ganyan ako gumawa ng question. Hindi ako magtatanong, this is where the growth of the microorganisms decline and they die. No, you will not see me ask you questions like that. Okay? You need to understand the principles of everything that I'm telling you, because in the exams, I'm not going to ask you a blank is defined as little or no multiplication. No, I will not ask you questions like that. Maybe one or two, yung mga hard to remember questions, but on exam day, you expect questions to be a little bit different. Okay? Next, in the stationary phase bacterial cycle, what are the probable reasons why the rate of bacterial multiplication ceases? Going back to the stationary phase, so it's at the peak, meaning it no longer goes up, it no longer goes down, it's constantly at a plateau, or it flatlines, top layer flatline, right? If there's a top layer flatline, the reason why there is a cessation of growth is because it's either the nutrients have been exhausted, toxic metabolites have accumulated, or development of unfavorable pH. Okay? So those are the things why the stationary phase of the bacterial growth cycles occur, or the bacterial growth cycle occurs. Okay? So yun. Very important si 95 kasi this one will not be asked about you. I won't ask this directly. But the idea with this is that you need to remember why stationary phase occurs. And these are the reasons. Nutrients have been exhausted, toxic metabolites have been exhausted, and that will go on when we talk about laboratory automation and laboratory microbiology testing. Okay? Now, what types of culture media? Now, we're talking about culture media now. Okay? So, I've mentioned culture media again and again, over and over it. Parang ang sakit sa ulo, sir. Pag-usapan na nga natin yung culture media. Right, Minty? Tigilan na natin ng ano. Ayoko na, sir. Puro kayo growth requirements. Let's go now to culture media. Okay? The third topic in our discussion. I think that this is the second to the last topic of this particular slideshow. All right, second to the last. The last one is infection control, which is another thing that is important. Okay? Now, what are the types of culture media based on state? Who can answer this? Physical state. Sige. Who can answer? What are the types of culture media based on physical state? Maria, Christina, Kay, Sabado.
Sir, solid, semi-solid, and liquid po.
Very good. All right. Now, differentiate the percentage of agar in each. 100% agar ba? Sobrang tigas nun. Gelatin. yun yung sago na kinakain natin. Alright? Ayun yung nasa boba. Alright? So, for solid media, you need only 2 to 3%. Okay? And for semi-solid, you need it at around 0.5 to 1%. So, kailangan talaga naming alalahanin yan. Oo. Magugulat kayo sa board exams ninyo. Tinatanong siya. Which of the following percentages indicate that a culture media is semi-solid? Mga ganong ano. Which of the following culture media is semi-solid? Mga ganong mga keme. Alright? So be mindful, okay? So again, remember your percentages. Okay? Now, what are the three basic types of liquid culture media? So you have 3 types of liquid culture media. Okay? So I'm not going to ask this to you guys. I'm not going to ask this because on your exams, because this is easy. You guys just need to remember there is a broth, there's an infusion, and milk media. Talaga, sir? May infusion pala. Ano yung ibig sabihin ng infusion, sir? So, basically, alam nyo na kung ano yung broth. Pakukuluan lang, di ba? So, you have, what do you call this? You have bone broth. You have what else? Meat broth. We have meat broth media, but that's for food microbiology. That's a different topic altogether. We have infusions. What are infusions? The most common example is brain heart infusion. So what happens is, what you do is, pinapakuluan din natin yung brain at saka yung heart. And what we do is, we mix it together. Kaya siya naging infusion. Alright? So another way of helping out when it comes to infusion is, alam niyo pag nagigisa kayo, what's the reason why nagigisa tayo? You want to infuse the flavors in the
Spice.
oil, 'di ba? Pag naggisa ka ng ano to, ng bawang, what you want to do is, you want to infuse acetylene into the oil to give it that garlicky smell. Allicin, sorry, not acetylene, allicin. So you want to infuse it. So that's basically what infusion is. An infusion is. It could be in a form of a liquid that is oil based, or it could be a liquid that is in a form of that's water based. So it depends on the type of infusion that you have. Okay? Next is milk media. Milk media, I just want you to remember, it's used for brucella. We have the bang bacillus. Bang bacillus, may mga lalaki na naman ang ngumingisi. Narinig lang yung bang bacillus, ngumisi na kaagad si Arnie. Arnie ha? Wag ganon. Arnie ha? Chill ka lang. Pag may milk media tayo, we have what you call the bang bacillus milk media. All right? It will turn violet. It will turn violet when you grow when there is a positive growth of brucella. Okay? So ano ba yung mga brucella? Kasi we're not going to discuss brucella that much. So this will be the first and the last time we're going to discuss brucella. So all you need to remember in terms of brucella is that you have three types, okay? Brucella abortus, melitensis, and bovis. Abortus. Okay? So those three brucella will grow in milk media. The only thing that you need to remember aside from brucella is that they cause what you call undulant fever. Undulant fever is you got infected, you're showing symptomology of your infection is that you have high fever, then it goes down, your fever goes down again, and then it goes up, down, up, down within a 30-day span. Kaya nga, we have what you call the brucella blood culture bottles. And these blood culture bottles, they usually have milk media in them or components of milk. Alright? Kaya important ang milk. Talaga, hindi lang pala sa tao. Yes, hindi lang pang tao ang milk media, okay? Kailangan yun din sila ng microorganisms. Now, most solid media are derived from the extract of what algae? Red algae, okay? Culturally grown, and they are grown in controlled environments so as to avoid contamination. Kasi in making culture media, especially solid culture media, you need to have a controlled environment. Otherwise, the organisms that you're growing might be contaminated with either substances or other microorganisms. So be mindful of a controlled situation. So common types of semi-solid media, it's very easy for you guys to remember. I think we discussed some of them. We have the motility media and the transport media. Okay? So they have a certain level of agar para hindi mabilis mag-dry. Kasi pag liquid yung binigay mo kaagad, magda-dry kaagad siya. The reason why we put a little bit of agar in our transport media is because we don't want it to be frozen entirely. We want it to have just a little bit of moisture. Some organisms require just a little bit of moisture. All right? Next, we need to talk about the two types of solid media. Well, it could either be liquefiable or non-liquefiable. Ano naman ang pinagkaiba nun, sir? This one has 2 to 3% agar. Ito yung niluluto ninyo. Nakapagluto na ba kayo ng agar before? Alright, very good. So it was part of your experimentation It was part of your experimentations when you were in your third years, I suppose. Correct? Tama ba, Clarice? Si Clarice kasi si Clarice kasi nakaganyan doon sa camera. Kaya sabi ko, okay. Parang inaantok na siya yung pinipilit lang niyang gumising. Kaya sabi ko, sige nga. I-call out ko nga si Clarice. Baka inaantok lang siya. Clarice, carry ka pa ha? Okay ka pa, Clarice? Ha? Carry ka lang?
I-take notes lang po.
Nakaganyan kasi si Clarice. Alright. Okay, so thank you so much for that wonderful talk. This is the first time I've heard you talk, Clarice. Wonderful. Alright. No, 0%. Bakit, sir? Meron palang agar na wala na 0%? Oh, meron palang media na walang agar? Oo. Yung mga karne. Okay? Umaya natin pag-usapan yon. So, examples of non-liquefiable media, chopped meat medium, rice grain medium, not really for bacteriology, but ayan, o di ba active na si James Paul? Nagising na din si James Paul. Kailangan lang pala manawag ng kaklase para magising. Alright, so we have rice media. Rice grain media is, yes.
Ay, sir, sorry. Nasa 2-hour mark na po ng lecture. So, pwede, after po nung kasabihin nyo, pwede na po kayo magpa-break.
Okay, so tapusin lang natin yung culture media. Carry lang.
Apo.
Tapusin lang natin yung topic. We'll move on to infection control. After we finish this one, we'll move on to infection control. And then that's the time where we're going to cap off. Okay? So, rice grain medium, not really much on bacteriology, but more so on fungal cultures. We're going to talk about rice grains when we move to mycology. Okay? So, let's move on to culture media based on composition. So, we either have chemically determined or chemically defined or synthetic medium, all right? Non-synthetic or complex medium, and of course, tissue media. Okay? What are the descriptions of each? Well, let me define to you guys what is a synthetic media. The exact composition is known, all right? So, exact composition. So, when we're talking about based on chemical composition, it's synthetic, okay? Meaning somebody created it for you. Meaning the exact compositions are known. There is a formula. Okay, a secret sauce, kumbaga. When you're going to the Krusty Krab, they have a secret sauce for the Krabby Patty. If you guys watch SpongeBob, I'm not sure if you guys still watch SpongeBob. There is a secret sauce for the Krabby Patty that's known as a synthetic or chemically defined product. Okay? A non-synthetic one is something that has one or more component that is not chemically known. Can you give me an example of a non-synthetic complex media or a complex media? Kanina kakasabi ko lang. Sige. Something that you don't have extensive knowledge with, but we still use it because it's proven to have grown certain organisms. Oh, nakalimutan na. Dinrawing ko pa kanina. Sige, James Paul.
Yes, sir.
Very good. Yes, very good. Very good. Why? Bakit James Paul? Bakit yeast?
Sir, madaming nutrient po.
Okay. And thank you so much. Yes, you're correct. Because one or more components is not chemically known. Alam mo ba lahat ng components ng yeast? May extensive knowledge ba? Kailangan ba dumaan ng manufacturer sa, kailangan bang isa-isahin nila ang components ng yeast? Siyempre hindi, di ba? What about blood? Do we know everything about the blood? We know some things, but we don't know everything about the blood. Essentially, we don't know everything about the blood. We may know 80% about the blood, right? We know that it's made of plasma, red blood cells, and everything. But do we know exactly what's inside the blood? That's the reason why you cannot replicate blood, diba? Up to now, we can synthesize, but we don't know the exact components of it. We have technology for that one. We can 3D print blood cells, but what's inside the blood cells is something that we still have no knowledge about. Or we still have little knowledge about, or maybe we're scratching the surface of. Alright? So, ganun yung dapat yung tandaan. Alright? So again, medtech question to. I'll be asking questions about this. If you're going to put a star on our slides, this probably may be the one that you need to. What about tissue media? So it's part of complex media, but this is an organism that this is used primarily for organisms that are obligate parasites. Okay? What do you mean obligate parasites, sir? I thought we are talking about microbiology, bacteriology, but we're talking about obligate parasites. That's parasitology, na, sir. You are confusing us. No, there are organisms that are obligate parasites. I'll give you an example. Treponema pallidum, right? Treponema pallidum cannot be grown on any synthetic medium. Okay? Leptospira interrogans cannot be grown on on any cell-free medium, right? Or cannot be grown on any cell-free medium. It requires tissues. All right? What else is there? Sige pa, ano pa? Sino pa? Wala na, nakalimutan na nila yung mga pinag-usapan namin last week. Oo, kayo talaga ha. Sige, yan ang mga itatanong ko kasi yun yung mga hindi nila alam eh. Di ba? Kasi ako gagawa ng exam ninyo. I was given the go signal by Sir Marco to give you guys an exam. I think November yata. I'm not sure. All right. So, most common examples of complex and non-synthetic media. This includes blood, plasma or serum, yeast and plant or animal extracts. Okay? Kasi hindi natin alam kung ano laman talaga nila. We may know some of the stuff, but we don't know 100% of the things. As opposed to synthetic, because there's a secret sauce. Alright? Now, examples of microorganisms that can grow on synthetic and non-synthetic media. They could be chlamydia, okay? Rickettsia, and viruses. Okay? Chlamydia, rickettsia. So don't forget those things. Bacteria din pala sila, sir. Yes, of course. When you in every book that you read about microbiology, specifically on the topic of chlamydia, the first sentence always say, these are gram-negative obligate aerobes, obligate parasites. Laging ganon. Lagi nyo makikita, obligate parasites. Pag nagbasa kayo ng ganyan. Rickettsia. Do you guys still remember your rickettsia? We have a specific discussion on rickettsia. Okay? Rickettsia subtropicalis, all right? Ehrlichia equi, these are organisms that belong to the family of Rickettsia. So I'm giving you guys an overview. I want you guys to at least try to have a background check on every organisms that we discussed. Okay? Obligate intracellular parasite. Very good. Thank you so much, Sir Marco. So, I'll give you guys an example of an obligate intracellular parasite. So, a great example is, yun nga, yung specialty ni Mark Segi. An obligate intracellular parasite can grow on solid medium. Okay? But, when it wants to become a pathogenic organism, all right, when it's infecting somebody, it cannot grow on your skin surface lang. 'Di ba? Kailan ka ba nakakita ng case na there is skin gonorrhea? There is none, right? So it requires the organism requires to go inside the cell and basically hijack what's inside. Right? So yon.
So, sorry sir ha. Kasi in my experience, when you say the term parasite, they think of ascaris.
Yeah. Kaya nga kanina sabi ko, 'di ba, Sir Marco, huwag kayong masyadong pa-comfortable kasi nga meron tayong mga organism, may mga bacteria tayo na pinag-uusapan na obligate intracellular parasites. Not just bacteria, we also have funguses. Histoplasma capsulatum is also known as an obligate intracellular parasite. So be mindful when you see the word obligate intracellular parasite, don't assume that it's the organisms that you talk about when you speak of parasitology. It could be in virology, it could be in mycology, or it could be in bacteriology. All right? Kasi madami ako nakita ng mga students. I think last time, Sir Marco, you showed me the statistics. 'Di ba? May sumagot doon na ang question is about gram negative, nakalagay na nga ang details gram negative cocci, Diplococcus, collected from the male genitalia, grown at 2% agar. Sorry, 2%, grown at chocolate agar plates after 2 days of incubation. Hockey puck colonies. Pero ang sinagot pa rin ni student is parasite pa rin. 'Di ba? Ang sinagot pa rin niya is Cryptosporidium ata 'yon. 'Di ba? Nandoon na lahat ng details, obviously bacteria siya. Pero ang inisip ni student, parasitology yung pinag-uusapan namin eh. Kasi pag nagpa-exam si Sir Marco, isang bagsakan 'yan, magkasama yung bacteria. Tama ba? Ganun ka ba magpa-exam, Sir Marco? Ay, tulog na naman siya. Tulog na naman siya. Sorry na. Basta ganun siya magpa-exam. Kasi I have access to Google Forms, to the Google Forms that he gives you guys. And pinagtatawanan namin kasi yung mga students. Charing. Charot lang. Charot. Hindi namin pinagtatawanan. Tinitingnan namin kung ano yung mga trends ng mga students kasi para on subsequent batches, we would know what to focus on. Kasi 'di ba nga, kaya nga ngayon minsan niya paki-clarify kung ano yung obligate intracellular parasite. Kasi nga last time may student na sumagot, nandun na lahat ng characteristics niya as a bacteria. Catalase positive, blah blah blah, superoxoid positive din, 'di ba? And then ang sinagot pa din niya parasite pa rin kasi nakita niya yung obligate intracellular parasite. Okay? So when you see this word, please don't assume that we're talking about a parasite. Okay? Okay ba yan? Okay, Arnie? Okay, Clarice? Okay ba, Minty? Ang ganda talaga ng setup ni Minty. It's very vintage, it's chic, it's just so madam. I love it. 'Di ba? Tapos may mga nakalagay, may mga bote dun sa likod no. Minty, alisin mo yung mga bote I'm assuming it's my God, ano yan, Minty? Ano ginagawa mo dyan? Bakit may mga oil-based products dyan? Kaloka.
Alcohol po yan, sir.
Okay. Akala ko kung ano yung mga oil-based products dyan sa likod, ha? Talagang kinuha niya para i-ano ko. Talagang kinuha niya para i-ano. Okay. Alright. I'm still putting you under, observation. Maybe the insides of the contents of that bottle could be something else. I don't know what it is. All right, the most commonly used tissue media in clinical microbiology. We're going to come back to that one. I said clinical microbiology, not bacteriology, because this may be the first and the last time we're going to speak about it. So might as well talk about it now. You have virology with me, so there will be an in-depth discussion of this one. So we have McCoy cells. Okay, these are from mouse fibroblasts. Vero cells from African green monkey. Kidney cells, okay, A549 from lung carcinoma, and HeLa cells from cervical carcinoma, and Hep2 cells from laryngeal carcinoma. Why do we use carcinoma cells? Who can answer this question? Why do we use carcinoma cells for tissue inoculation? Sige. Who can answer? Why do we use carcinoma cells? Because carcinoma cells are what? What? Sino makasagot? Naku, mahirapan tayong mag-break nito. Walang makasagot. Ano ba ang Ayan na. Yan na nga ba sinasabi ko. Bright na, pretty pa. Nag-blush on pa to. Sandali, tingnan ko. Nag-blush on pa to si girl. Talagang ready talaga siya lagi kapag magsagot. Sige, smart na, pretty pa. Okay. Nigel.
Try ko lang po. Rapidly dividing.
Okay. That's fine. They're rapidly dividing, but something else. What's the main difference between normal cells and carcinoma cells? Histopathology review. Grabe, holistic talaga ang approach ni Sir. 'Di ba? May histopathology pa. 'Di ba? Ayaw ko tinuturo yung histopathology, pero 'di ba? Wala lang. Sige, why do we use carcinoma cells for tissue cultivation of microorganisms? Sige. Naku, mukhang magbibigay ako ng price ulit ngayon. Hindi sila motivated. Sige, for 100 Gcash points. Wala ba si Diandra, Sir Marco? Wala ata si Diandra. Sige. Who can answer? Come on, guys. Sige, Arnie.
Sir, try ko lang din. Um, yung cancer cells po, instead of dying, they multiply po and produces abnormal cells.
All right. What's the term that I'm looking for? Not die. They don't die. They constantly divide. Essentially, they are what? Nandun na from the definition that you've given me. They are what? Wala na. Siret na. Magbibigay ako ng 100. They are what? I'm looking for a specific word. I am looking for a specific word, children. Children of the corn. Alright? They are 5, 4. O, Nigel, sige, go.
Malignant?
Wrong. Sorry. They are 5, 4, 3, 2. Sino yun last? Hindi nakaabot si... Sige, Maria Cristina. Okay, Sabado.
Apoptosis.
Apoptosis is death of the, is basically programmed cell death. Nandun na nga sa definition ni Arnie kanina na natumbok na niya pero hindi niya talaga masabi sa akin kung ano yung ano. Hindi lang maganda yung execution, Arnie, but you had the idea. They are, kakantahan ko kayo, they are immortal. Diba? They are immortal. They don't die. Diba? Kaya, di ba namamatay ba ang carcinoma cells? Lagyan mo lang ng proper nutrients yan. Continuous ang division yan. Right? Because they have damaged or they have damaged or they have a mutated telomerase structure. Meaning, pag sinabing telomerase, yun yung responsible for capping off. Ito na yung last time mo na magdi-divide ha. Pag wala ka na kasing telomeres, yung cells mo, pag wala ng telomeres, hindi na siya mag-divide. Hence going programmed cell death. Yung yung sinasabi ni Maria Cristina Sabado, apoptosis. So pag pag meron kang mga genes na may mga cell, not all carcinoma cells are like this, by the way. They don't have telomerase. Okay, but majority of there's a problem with the telomeres. Okay, it could be a problem with the structure itself. It could be a problem with the enzymes for telomerase. Okay, so what happens is that these cells continuously divide with proper nutrients, with a constant supply of nutrients. Kaya ito, kinukulture lang yan paulit-ulit. Okay, yung HeLa cells actually galing sa madre yan. Alright, sa cervical cancer na isang madre. And I actually have a link for you guys to watch later on. Okay? HeLa cells later. I think ewan ko kung interesting sa inyo pero just to entice you guys in reading about on studying about HeLa cells. Okay? So maganda siyang introduction for biology students, much like us. We are all of us here are scientists. So you guys need to watch. You can watch this, guys. This is known as this is the story of where or how HeLa cells were developed. Okay? So that's a YouTube video for you guys from TED Talk. It's a short video. Don't worry about it. Anyway, you can watch it afterwards. Types of media based on dispension and distribution. So we can either have a tube medium or we can have a plated medium. What's the difference between the two? Well, it depends on how you sterilize it, okay, or dispense it. So what you do first is you weigh, dissolve, sterilize, and dispense when it's plated. But it's different when it's tubed. You dispense first and then you sterilize. Why? Kasi flat yung surface, and usually the agar is liquid pa. Kaya kailangan mo siyang basic common sense actually occurs here. Maglalagay ka ba kaagad na i-sterilize mo ba yung petri dish? Pupunuin mo yung pupunuin mo yung autoclave mo ng petri dish. Hindi, 'di ba? Ang ilalagay mo yung Erlenmeyer flask. 'Di ba? Sa autoclave bago mo siya i-dispense. Whilst on tube media, dispense mo na. Kasi hindi naman yan nagiging solid or liquid eh. Hindi din natutunaw ang petri dish mo. Although may glass na petri dish, but would you rather alangang isa-isahin mo yun pag aalsin mo sa culture sa autoclave, 'di ba? Basic common sense applies here. All right? But again, this is a question that was asked in one of the board exam recall questions. I remember it was, I think, 2013, when one of my interns, she came back to me in the hospital and said, "You know what, sir? One of the questions that you asked me whilst I was an intern under you was asking the exams. What's the order of dispensing tubed media?" Binabali-baliktad lang nila. And then dito siya nung nakita niya na yung question na weigh, dissolve, sterilize or dispense, dito na nagkagulo sa third and last. Kasi basic common kailangan talaga minsan nagtatrabaho ka sa laboratory kasi hindi mo siya maaalala. Kaya lagi kong ini-emphasize sa students to, at saka even my interns when I was working in the Philippines, that you have to be able to do laboratory work in order for you to appreciate what is being asked of you in the exams. 'Di ba? So nakita mo, alangan kunin mo isa-isa yung in-sterilize mo yung kung inuna mong i-dispense, alangan nag-dispense ka na ng blood agar, 'di ba? I-dispense mo pa yung blood agar, hindi naging chocolate na yun. Edi kung inoutoclave mo yun, hindi naging chocolate yun, 'di ba? Nagets nyo? Nagets nyo? Parang ganun, para mas madali nyo maintindihan or madali nyo maalala. Pag in-sterilize ko kaagad yung agar, nag-dispense na ako ng blood agar. Ng blood. Di pag nilagay ko sa autoclave yun, magiging chocolate yun. 'Di ba? Or alangan kunin ko isa-isa yung plates na may laman na agar doon. 'Di ba? Di lalo akong mahihirapan. Tsaka another reason is may pressure 'yon, 'di ba? Mababasag yung magkakaroon ng uka-uka yung agar mo. Para madali nyo lang maalala. Although hindi naman talaga nangyayari. Kasi nobody is that thick who would say, dispense ko muna sa ako ilagay yung mga media dun sa sa autoclave. Wala namang ganong tao. Wala pa akong nakita na ganon. Alright? Pero kasi kapag hindi kayo hindi kayo nakapag-practice ng ginagawa sa lab, baka makalimutan nyo ba? 'Di ba? At least ngayon naalala niyo na yung mga tube lang ang ilalagay niyo muna, i-dispense niyo muna before niyo i-display, before niyo i-sterilize. Okay? Autoclave when I say autoclave, when I say sterilization, it means autoclaving. Now enumerate the types of media based on function. So there's 8. Let's go one by one before we go to break. Malapit na tayo mag-break. Simple or basal media, enriched media, enrichment media, selective media, differential media, transport media, media for susceptibility testing, okay? And media for biochemical testing. So the last two is going to be very important for you for your discussion because most of my questions usually come from this. This one is basic knowledge that you must know. Okay, transport media is also important, but specifically for virology. So let's talk about media based on function used for supporting the growth of non-fastidious organisms. So these are simple media or basal media. Example of that one is your nutrient agar, right, or trypticase soy broth. Okay, TSB or TSA. If it's TSA or it's TSA, it's solid. If it's TSB, it's a broth, meaning it's liquid based. Okay, so sometimes you might be asked a question. Trypticase soy broth is what type of media? Solid, semi-solid, agar, or liquid or tissue media. Diba? Lilituhin lang kayo. Alright? Media type based on function that supports the growth of fastidious organism. This is known as enriched media. Common examples, of course, is your blood agar and your chocolate agar. Okay? BAP or CAP. What is in blood agar? Contents of blood, of course. What about chocolate agar? Hemin, right? The byproducts of degraded hemoglobin. A lot of microorganisms love it, particularly the members of the HASEC group, Neisseria and Neisseria. Okay? So be mindful of these things. Why do we need them? Because they are enriched media. In the laboratory, majority of the time you will be plating your culture. You will be plating your cultures not in nutrient agar, but mostly in blood agar plates, chocolate agar plates, and makonki. But you'll know later why. Okay. So next, What's the most commonly used blood for blood agar? Indicate the calculation and the percentages. Okay? Or the calculated percentage. So you are going to use defibrinated sheep's blood. Okay? At about 5%. Remember the percentages. How do we do defibrination? That's the next question. Or why is human blood not preferred pala muna in preparing BAP? Bakit sheep's blood, sir? Grabe, bakit kailangan sa tupa? Kawawa naman yung mga tupa. 'Di ba? Bakit? Kawawa naman, Nigel. Bakit kailangan? Kasi pag kumuha ka ng sa tao, hindi natin alam baka mamaya nag-antibiotics yung tao, 'di ba? 'Di ba? Hindi natin alam baka mamaya ang pasyente may other disease that causes an increased, 'di ba? Intern's blood is actually used sometimes. Yes. Yes, intern's blood for merits katulad ng mga pala-absent. Feeling ko si Minty isa siya doon sa mga pala-absent. Pala-absent ba yan, Sir Marco? Si Minty? Si Valiant?
Actually, I didn't know na sumasagot pala siya until seminar 1. Tahimik lang yan.
Syempre kasi bakla yung nagsasalita eh. Syempre, we resonate with each other. Kumbaga sa Genshin Impact, meron kaming liquid resonance. 'Di ba? Nasa dugo 'yon. Parehas kaming dendro element kasi parehas kaming green ang dugo. Ganon. Kaya nagre-react siya ngayon. So hindi siya pala absent naman, sir. Alright? So at least kahit papano pwedeng gamitin yung blood ni Valiant. Okay? Pwedeng gamitin yung blood ni Valiant. Kapag ka routine lang yan. Pero ideally, in the books, okay? Walang mag-e-epal sa ospital ha, please. Marami ako nakikita mga naging intern dati, masyadong nagpapakyut sa mga professor, sa mga staff, na busy sila, masyado silang busy, wala na silang pakialam doon sa theoretical knowledge. Okay? Walang babanat ng ganto. Eh di ba sir, kailangan sheep's blood? Bruha ka, nandun ka nagtatrabaho ka sa public hospital, ang dami ng samples, tapos iisipin mo pa maghanap ka ng intern blood na lang. 'Di ba? Ganon. O kaya yung mga i-discard na blood bags ng ng mga donors natin. 'Di ba? Wag ganon, walang magaganon sa ospital ha. 'Di ba? Holistic talaga ang approach ko, Sir Marco. May mga life's lesson pa kasi may mga student kasi ako dati. May mga student kasi kami dati na 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 nagagaganyan dun sa ospital, nabubwisit yung mga senior ko. Ako kasi medyo ako eh. Siguro ngayon hindi na ako pwede ngayon. Madali na akong mabwisit. Dati kasi bata-bata pa ako eh. Siguro ngayon medyo madali na din ako mabwisit. Kapag may mga tanong, di ba sir? Di ba sir dapat na gagamitin natin? Di ba sir dapat hindi cold ang na hindi nakalagay sa ref kapag gagawa ng smear? Nabubwisit, busy, toxic na toxic, 100 ang samples, tas dengue season pa. Tas magagaganon ka, talagang mabubwisit sa inyo ang staff ninyo, ha? Walang magmamaganda ng ganun sa ha? Sa clinical rotations, ha? Chill lang, yung nakaupo ka lang doon, pag inutusan ka maggawa ng cotton balls, gawa ka ng cotton balls. Matutuwa sa'yo si sir or si ma'am. 'Di ba? Ganon. Ganon kaya ang lecturer nyo ngayon, passed with flying colors without merits and without demerits, 'di ba? Ganon dapat ang pagpapakyut, hindi yung pagpapakyut na nagpapakatali-talinuhan kayo, ganon. Wag ganon. Mapapagalitan kayo doon. Okay? All right. So blood is not preferably used because it has non-specific inhibitors. Yun nga, maybe the person is taking antibiotics or maybe nagtitake ng alaksan, baka mamaya nagkukos pala ng false growth or non-growth of organisms yon. Okay? Next, what's the effect of certain substances? So we have citrate and dextrose. Citrate inhibits the growth of Streptococcus species, whilst dextrose dextrose alters the hemolytic patterns. Kaya nga 'di ba, citrate, kaya hindi masyadong ginagamit ang blood bags. Kasi ayaw talaga namin. But kasi nga wala nga theoretically you really shouldn't use blood bags in preparing your blood agar plates, but because of the lack of funds, we still opt to use it. But just remember, maybe streptococcus 'yon, 'di ba? Lalo na kung vaginal swab ang ginamit, kasi important ating ma-analyze ang group B strep sa vaginal swab kasi it can cause spontaneous abortions. Okay? Dinidetect yan sa vaginal swab. So dextrose din, Okay, hindi din pwede kasi it alters the hemolytic patterns. Before we go on to hemolytic patterns later, what are the 3 types of hemolytic patterns nga? Who can answer? Sige, ni gel na naka-blush tsaka naka-lipstick.
Alpha, beta, and gamma.
Okay, define each.
Hemolytic.
Siyempre, nagsabi ka na eh. Siyempre, alangang bitinin kita. Sige na, go. Go.
Pag alpha, incomplete hemolysis. Pag beta, complete hemolysis, yung may clear zone. And pag gamma, wala.
Alright, good.
Complete naman.
Bakit ganun ka lagi talaga sumagot, no? Laging may hesitation. Laging merong 3 to 5 second pause. 'Di ba? Obviously, pag incomplete yung isa, complete yung isa. 'Di ba? Ikaw talaga ha. Niloloko mo talaga ako. Kanina ka pa. Tinanong kita kanina, ganyan din yung ano mo, 'di ba? yung isa gram positive. Si Listeria gram positive. Tapos si Yersinia 5-second pause. Gram negative. Ganyan. Siyempre, ikaw talaga pag mga opposing ideas talaga meron. Parang medyo I think ang problem ni Nigel is not that she's not knowledgeable, it's that she lacks the confidence in answering. 'Di ba? So main concern ko sa iyo is the confidence. Okay? So confident dapat tayo ha pag sumagot. Pagka mag-alala, hindi naman graded ang confidence mo, pero at least in terms of discussion, you have an idea. Now,
Sir, that's not true.
Really?
Kay Nigel Berongoy kasi student ko siya from Micro.
Oh, really?
So they have video submissions. Production value yung batang yan.
Totoo? Bakit pagka mga related pag mga related lang pala sa academics, pero pag mga kantahan, mga ganon, yung mga sayawan, yung
Production value yung batang yan.
Oo, yung magi-split, Sir Marco. Mga ganon.
Habang nag-i-streak ako,
Yung
naka-split.
mga ganon pa. Diba? Valiant, kailangan mo talagang talunin to si ano. Yan ang competition mo. Ha? Anak. Talagang tinawag ko na talagang anak. Anak, ha? Anak. Ayan ang kailangan mo anuhan. Kailangan mas mataas lagi ang mga grades mo dyan, ha? Anak. Magagalit ako sa iyo, hindi kita bibigyan ng allowance kapag hindi mataas ang grades mo sa kanya, ha? Anak. Talagang ginatawag ko sa Aya. Hindi na minty. Anak. Alright. Okay, enough na tayo dyan. Blood from what animal is preferred when isolating species of Haemophilus? So again, ideally it's horse. Okay, horse blood. Because for differentiation, or defibrinated for differentiation. The species of Haemophilus that can be used in this particular case is the beta hemolytic Haemophilus, such as Haemophilus parahemolyticus and Haemophilus hemolyticus. So beta hemolytic Haemophilus can be used to differentiate them. So they're basically observed to have beta hemolysis rather. Okay, so isolating. Or theoretically, but you can still isolate Haemophilus in intern's blood. We've done that before, but theoretically, the ideal blood source would come from horses. Okay. Now, in preparing CAP, what temperature would be ideal to heat blood? 80 degrees Celsius. Okay, so correct. Naalala pa ni Sir Manuel. And they're useful for the isolation of Strep pyogenes, Haemophilus species, and Neisseria species. We're going to talk about it some more when we go to the specifics of the microorganisms. But for today's lecture, we will just scratch the surface. Okay, just a basic review, by the way. Now, what media is used to enhance the growth of a particular organism? So this is enrichment, different from enriched. Okay, enriched may past tense. Ibig sabihin, nandyan na. Enrichment, noun. Okay? So, di ba, yun lang yung tatandaan ninyo. Pag sinabing enriched, past tense siya. Okay? Past tense. Enrichment, iba ang prefix, but it functions as a noun. Oh, di ba? Ang galing-galing talaga ni Sir magturo sa inyo ng mga kagulo para mabilis yung maalala. Pag enriched, past tense na, nandun na kasing enriched. yung enrichment molecules. Okay? Pag enrichment, yung organism ang ini-enrich natin. Okay? Nakita nyo yung difference? 'Di ba? Clear ba, guys? 'Di ba ang galing-galing, ang dali-daling alalahanin ng enrich. Past tense nga ibig sabihin nandun na yung mga enrichment molecules like yung mga hemine nga na mga sinasabi ko, 'di ba? Ganon. Okay? So, they are what you call this they are these organisms grow best let's talk about the example of media, by the way. They grow best in the following: tetrathionate broth for salmonella species, selenite broth also for salmonella and shigella species, and alkaline peptone water. Alright, bakit siya ini-enrich? Kasi nga alkaline to, 8 to 10 ang pH. Ito may tetrathionate or may selenite. 'Di ba? They thrive in these things. These organisms, they grow better. Instead na onti lang yung colonies, sila yung mas madami. Not saying not saying that it will cancel out the other organisms, but they would thrive better. Sila yung mas madami ang tutubo. Gets nyo? Alright, so ilalagay ko yung pupo, for example, ng pasyente sa selenite broth. Will it inhibit the other organisms? No, it will it will slow down the process, the metabolism of the other organisms, but it will enrich the growth of salmonella. Therefore, it's easier to isolate the organisms. Okay? Alkaline peptone water, it doesn't necessarily destroy the other organisms. It can destroy the other organisms if it's halophilic, if it's acidophilic, but not necessarily destroying the other organisms. But when you treat the subcultured organisms or the subcultured bacterial suspension, you will see it's better. There is better, there's better growth or there's better isolation. Okay? Gets nyo? Gets nyo? Hindi niya pinapatay. That's a different one. Okay? That's a different type of culture media. Okay? Next. Now, media type based on function that is used to grow a particular organism by using inhibitors. That's now known as a selective medium. Now, it's different from an enrichment media which supports the growth of these organisms. A selective medium, particular organism ang tinatanggal niya. Ang pinapatubo niya. Gets nyo? It grows by using inhibitors. Okay? So examples of inhibitors used in culture media, we have crystal violet or gentian violet. It's used in usually we can use dyes, or we could use a bile salt, or a salt, sodium desoxycholate for certain organisms like a specific one that we're going to talk about later. Okay, like bile esculin. All right. So we're going to talk about that later. All right. So inhibitors for gram-negative bacteria. For gram-negative bacteria, we have sodium azide or potassium tellurite. So these are just general examples. Okay. So dyes, you could use crystal violet or gentian violet, or you could use bile salts like sodium desoxycholate. Now, if you want to inhibit gram-negative bacteria, you either need sodium azide or potassium tellurite. Okay? Another one that we can use, a common example of selective medium for Mycobacterium tuberculosis is Lowenstein-Jensen agar. Okay, the inhibitor that we use there is malachite green. Kaya nga green ang ating MTBA. Hindi ko sure kung pag ginugel natin ganun pa rin yung itsura na. And then Much granules are seen on the AFB stain. Usually, if it's direct sputum analysis, you wouldn't see much granules. But when it is grown, or it's an organism that has been grown in Lowenstein-Jensen, you will see much granules. So let me show you what Lowenstein-Jensen looks like. I'm not sure if you guys are familiar with the. Let me Google search it for you. Alright, so makikita nyo, green siya malachite green. So tatanungin nyo, which of the following dyes inhibits the growth of other organisms to support the growth of Mycobacterium tuberculosis? Diba? Yan yung makikita nyo. Cauliflower colonies, Don't forget. Okay? Diba, diba? Easy? Easy to remember? Diba? Kasi pag hindi nyo nakita, hindi nyo maaalala, diba? Diba, diba? Kaya nga meron tayong tinatawag na monkey see, monkey do na learning methodologies. Okay, now selective medium for Corynebacterium dipterae and the inhibitor used. For Corynebacterium, we have what you call Muller's tellurite medium. Remember, the organism is selective. The media is selective for a gram-positive organism, diba? And the inhibitor that we're going to use is obviously something that will prevent the growth of gram-negative organisms. Therefore, we're using tellurite or potassium tellurite. Gets nyo? Gets nyo na kung anong function ng, ng mga ganyan? Diba? So, when I ask you the question next time, diba? Which of the following is what is the action of potassium tellurite in Muller's tellurite agar? Hindi ko sasabihin yung organism kasi question number 2 yon, di ba? Hindi ko sasabihin yung organism. Question number 2 related to question number sa ako yung natatanong. Which of the following is the action? Is it a color indicator? Is it an inhibitor? Is it an antibiotic for inhibition? Or is it an antibiotic for supporting the growth of the organism? Or is it a naturally occurring substance when preparing the agar? Obviously, you're going to select it's an inhibitor. Next question. What is grown in Muller's tellurite agar? Or Muller's tellurite agar is selective for which microorganism? Yun. Di ba ganun magtanong si sir? Pag hindi mo naalala yung isa, mali na lahat. Di ba? Ganun. Ganun ako magpahirap ng students. Di ba? At least meron kayong idea. Di ba, Sir Marco? Okay. Next. Selective media used for Neisseria gonorrhoeae. This is actually you need to put a star on 121 because many of my students would come back and say that they were asked this question. Which of the following is not media used for isolation of gonorrhoeae? Which of the following culture media is ideal for Neisseria gonorrhoeae? And most of the time, these 4 will come into your question banks. Okay? Thayer Martin, Martin Lewis, Modified Thayer Martin, and NYC agar. Ano yung NYC agar, sir? New York City agar lang siya. Kasi it was discovered in New York City in the 1980s. 1970s or 1980s. Okay? Now, selective differential media for Staphylococcus. When we talk about Staphylococcus in specifics, we're going to talk about mannitol salt agar. I think I mentioned this before in our early discussion. So just remember, mannitol salt agar contains mannitol as your sugar content. This is what the organism metabolizes. The inhibitor is 7.5 NaCl, making it a halophilic culture media. And lastly is the phenol red indicator. So, ano ang organism natin? Ano ang mag-grow? Anong color ng organism sa mag-grow? Red colonies. Sorry, yellow colonies. Kasi, pag kinain ni Staph specifically Staph aureus yung mannitol, it will create an acidic reaction with phenol red, and the acidic color of phenol red is red. The original color of phenol red is kind of reddish, sometimes pinkish, depending on the concentration of phenol red in your stock reagent. But usually it will color yellow. It will create a color yellow rather. Okay? So let me show you the colors of Staph aureus. Staph aureus in MSA. It would look something like this. So we have this in the laboratory. But unfortunately, I can't take out the agars, and I wouldn't risk taking it out of the laboratory also. So it will look something like this. So phenol red nga siya. So sometimes it's pinkish, and then when the organism metabolizes the mannitol, it will create this kind of color. Okay? Kasi nga naging acidic. Alkaline, naging acidic yung environment. Okay? Gets ba? Gets? Nakaka-follow pa, guys?
Yes po.
Na. Alright. Malapit na tayong matapos. Mamaya magbe-break na tayo. Alright, so in MSA, so we already know the answer to this one. Fermenter. Mannitol fermenter is differentiated from a non-mannitol fermenter by what? The color yellow. Okay, non-mannitol fermenter is a pink colony. So usually, it's Staph aureus that will be a mannitol fermenter. The other ones are non-mannitol fermenters. Okay? So sometimes I would receive samples from the ICU, like axilla swab, what else? Nasal swab, and I think groin swabs also. Alright? So, anong purpose? They want to detect staph. Really? Bakit? Anong purpose? Di ba lagi naman talaga tayong may staph? No. The purpose is they want to know if the patient has methicillin-resistant staph aureus or MRSA. Alright? So, it's important to do that. So, usually we just want to grow the staph, and we don't use blood agar anymore. We just want to screen if there's staph. And if there's staph, we want to do identification. We want to do identify. We don't usually go with identification because we know already that it's staph. But if the colonies look a little bit funny, we'll do identification. We'll still do identification, but automatically we'll just use a kit to determine if the Staph aureus colonies that grew were actually MRSA. So ganun yung ginagawa namin sa laboratory. We just get the swabs, plant the organism. By means of planting, I mean, when I say planting, I meant inoculating. We just inoculate the swabs in MSA, and then that's it. Because we're not just doing culture and sensitivity, okay? We're also doing culture. Sometimes you would see MDRO screen in the requests. So MDRO screen basically allows us to gives us doesn't give us a lot of leeway. You just need to check if there is a specific organism. So we use selective media for that one. In this case, it's Staph aureus. Okay? Do you guys still follow? Gutom na? Mamaya magtatanong ako ng mga ganyan ha. Huwag kayong etchusera. Novobiocin resistance test is another thing that can be used to differentiate the non-mannitol fermenting microorganisms. We're not going to talk about this, but just for too much. But just for your information, Staph epidermidis is sensitive to novobiocin, while the other one would test positive, which is of course Staphylococcus saprophyticus. Novobiocin resistant or positive for novobiocin resistance is your saprophyticus. Okay? All right. Now, differential media for Vibrio species is your TCBS, or what you call your thiosulfate citrate bile salt sucrose. The pH indicator is, of course, bromthymol blue. All right. And the sugar that you use is sucrose. Okay? Sucrose. Bromthymol blue. What's the color of bromthymol blue? The inert color of bromthymol blue. It's green. It only turns blue when there is a fermenter. Okay? Next, in TCBS, the colonial appearance of a sucrose fermenter um, is that they are like this. A sucrose fermenter will have yellow colonies, and a non-sucrose fermenter will have green colonies. Okay? Example of differential media for gram-negative enteric bacteria. So we have EMB, McConkey. What's HEA and what is SSA? Who can answer HEA? Does anybody know what HEA mean? Sige nga. James Paul.
Ectowin, enteric,
Okay.
agar.
All right. Very good. What about SSA? Basic knowledge. All right. Alexis. Alexis, where are you, Alexis? Alexa, answer the second one. Charing. O sige, ayan na si Mary Kay. Ang tagal ni si McKay na muna. McKay?
Salmonella sugar po.
All right, very good. What about EMB, the last one? EMB. Si McKay ulit. Okay, McKay, what's EMB?
Eosin methylene blue.
All right. Thank you so much. Very good. Okay. So, we'll move on to transport medium. Okay, so transport medium, I think this is the last topic that we're going to discuss. Transport medium, we have four that you need to remember. Okay, Amy's, Carrie Blair, Stewart, and Transgrow. And based on composition, what do all these media have in common? They are all semi-solid. Remember what I told you guys earlier? If it's for bacterial transport, it needs to have a little amount of moisture, not so much wetness. Okay? If it's for viral culture media, that's the time that you'll have a full-on liquid culture media. But most of the time for bacterial culture, you just need to have a moist environment for them to grow. Okay, now, component of all transport media absorbs fatty acids and other toxic substances. Usually, it's charcoal. That's why you tend to see black specks. I'm not sure they're in the Philippines because when I was working there in the Philippines, the gel of Carrie Blair, or sorry, Stewart, we were using Stewart back then, was I think it was clear, so I'm not sure, but here in the Middle East we use charcoal, so I can see it has black specks. Okay. Next, so we're talking about susceptibility testing next, and I think that is the end of our discussion for nutrition or bacterial nutrition, 'di ba? Pati pala yung bacteria may nutrition, sir. All right. So that's the end of our discussion for nutrition. I'm giving you guys like 20 minutes because I need to eat lunch also. I think it's lunchtime here. Okay? 20 minutes only. We'll reconvene after my timer. So I'll leave the timer here. Let me just stop